registration at the
front.
front desk is going to assist them.
Okay. It's like a step ladder. Yeah. Good morning, colleagues. We're just going to get started. I think we're already running about 15 minutes behind time, and we don't want to steal precious minutes from the exciting presentations that we will receive from our various colleagues this morning. But good morning again. My name is Tshwanelo Rakaibe, and I'm a senior researcher at the Energy Center, and I'll be the MC for the first part of the program this morning. But before we get into that, I'd just like to welcome Chris from the ICC to do the safety briefing.
Good morning, ladies and gentlemen, and a warm welcome to the CSIR International Convention Center. I'm VP Pretorius, Safety and Setup Coordinator of the Center. Before you proceed, I'm just going to give you a couple of health protocols as well as evacuation protocols of the Center. Kindly make use of the hand sanitizer stations located within the venues as well as the communal areas as frequently as possible. Should you fall ill or require medical attention, kindly report to the reception area whereby a first aid facility as well as trained responders will be made available to you. In the unlikely event of an emergency, an alarm will sound throughout the building. Trained responders will give instructions on evacuation. Stay calm. Take only belongings that you can carry. Walk in an orderly manner. Do not run. The responders will lead you to an emergency exit, then to the assembly point. Restrooms are available throughout the building. Please look out for signage. As this is a public building, please do not leave your valuables unattended. Free wireless internet access is available throughout the building. Please select free CSR ICC on your device. No password is required. For assistance, please call on any of our staff members who will contact the technician to provide support. Thank you and enjoy your session.
Thank you, Chris. I hope everyone knows what to do if there is an emergency. But really, I think I have the easiest job today here at the symposium, just welcoming our esteemed speakers onto the stage. I think we're all really excited to hear the different presentations that we'll receive on the exciting work that's being done by our different colleagues in the different clusters and divisions in the CSIR. We're going to have 3 sessions during the course of the day. All of them will be in this venue. We'll start with a health and biomedical session, which will be chaired by Dr. Nobuhle Majozi, which would start around 9:15 after the opening remarks. And then we'll have a break around 10:15, where there are some posters that are on display. Please do engage with the content of the posters, speak to the researchers to find out what it is that they've been working on. And then we'll have a second session, also a continuation on health and biomedical, which will be chaired by Dr. Cindy Swabutelezi, starting around 11:15. And then we'll have lunch between 12:15 and 1:20. Then we'll have the afternoon shift. I hope we're all still awake and energized for that session. I know it's usually termed the graveyard session because we're all full and have heard numerous presentations at that point in the day. But this will be the biotech and energy session, which will be chaired by Yonela Tukwayo, and that will take us to about 3:20. And then there'll be a last opportunity for all of us to engage again with the posters that are in the exhibition area outside in the foyer. So without wasting any further time, I'd like to call on Dr. Tshepo Tsekwa, who's the Chief Researcher here at the CSIR, to do the opening remarks for us.
I don't know if I need this. I hope you can see me without it. You can't see. That's very rare because I think I've been on this podium a few times. So thank you very much, Sanelo, for the introduction, and thanks for the invitation. Good morning, everyone, and welcome. So I was on the agenda as far as I knew. Nokwanda and Pinda as for welcoming remarks, but I see now it's a plenary address. So I'll stretch it out a bit, but don't be too alarmed by what I'm about to tell you, and don't be too shocked. But we'll get there, and you'll realize that in actual fact I'm not being too disruptive or naughty. But I'll start by telling you that almost 18 years ago to the day, I think I started at the CSIR on the 1st of September in 2007. And now I was at the time a slightly smaller, maybe narrower postdoc in the old biosciences unit. And at the time, postdocs were actually designated candidate researchers. So you can imagine, at the time I arrived, I think I had a total of 3 papers to my name, and so I think I had no clue what a technology demonstrator, the famous output from Mary Naudé, is. I had no clue, like many of us coming from academia, what TRL stands for, and so I could say that at the time I was very firmly and unambiguously an emerging researcher of the CSIR, like many of you here today. And so the very next year, maybe the next year or two, the very first Emerging Researcher Symposium was actually launched, and I remember very well participating. I gave a presentation on plasmodium proteomics. I used to work where a couple of my colleagues here are working now in the proteomics area of the CSIR. And then a year later, I also then participated as part of the committee reviewing some of the submissions and also awarding posters. So you can imagine that I'm very pleased and very honored, and it's quite a pleasure having climbed through the ranks from firmly an emerging researcher to be now giving these opening remarks. It's listed as a plenary, it's not, it's just a few opening remarks just to set the scene. It's really an honor having climbed through the ranks as a chief researcher to be here, and I hope you'll appreciate the few as I have. So I did say at the beginning that you shouldn't be too alarmed, and I think that the HC colleagues who invited me will excuse the one word I'll use in my introduction, and I ask also for Tsanelo's apologies as the MC for what I'm about to say. The point is that in the 17 years I've been at the CSIR, I've learned the subtle art of not giving a... Can I say it? I'll say it only once and then I'll tone it down. I've learned the subtle art. It's going to get me in trouble. The subtle art of not giving a fuck. And I'll say it only once, and I apologize, my colleagues. I can see, please don't be too alarmed. I'm going to call it not giving an F from now on, just so that, I've been naughty once, and I'll leave it at that. But the remarks are not really about laziness or apathy. They're not about you not caring at all about your work. What I wanted to say is really about focus, values, and choosing very precisely where to invest your limited time and energy and your limited passion as an emerging researcher at the CSIR. So it's a lesson I found powerfully captured. I could realize as I was saying what I was saying that some of you might have read the book that inspired my remarks this morning. There's a very nice book I read recently that talks, and it's titled The Subtle Art of Not Giving an F. And it's quite a powerful book, and as I read it, you realize that some of the tips in there, some of the guidance is very relevant to what we do as researchers, and certainly as emerging researchers at the CSIR. So the book almost perfectly sums up what research, development, and innovation at the CSIR is about and what it teaches us every day. There are 3 lessons. The book is quite extensive with many different chapters, but there are 3 lessons that capture, in essence, what I believe we can learn from not giving an F. So the first lesson, and I've alluded to this already, is that it's very, very important to choose what to care about. When I was an emerging researcher that many 17 or 18 years ago, it was the inclination is to care about everything, to want to do everything. You want every experiment to work, every reviewer to like your paper, every proposal to get funded. I spoke about the TDs. You want every TD to be accepted by the elusive technology demonstrator panel of the CSIR. And you tend to believe that that's what serious scientists must do, and that's what it must take to climb the ladder. But, like in life, if you care about too many people, or if you care about everything, at work, you end up quite literally caring about nothing. You spread too thin. You chase every opportunity, stress about every criticism that could be viewed as feedback. And truly speaking, it's not productive to chase every opportunity, because you burn out before you even hit your stride. So it's very, very important that you follow the subtle art of choosing carefully which problems are worth solving, which collaborations are worth building, which sacrifices are worth making. And you'll always have problems in R&D and in research. It's actually the nature of the job, and the issue is not for you to avoid those problems. It's for you to direct your attention and passion to solving the right ones. So the bottom line here is that stop giving an F, and I'll let you say it in your mind because I'm going to get in more trouble if I keep repeating these words. Stop giving an F about everything and start giving attention to the right things. The second lesson, and they're all interrelated in some ways. The second lesson is around redefining success. And if one had been told this as a 29-year-old researcher just starting out at the CSIR, success, even on the career ladders that we pursue so much, is not necessarily about the number of committees you sit on, how many acronyms you can decode, etc. It's probably more about aligning your work with what values, with values that matter. Values that matter to you, values that matter to the community around you, values that matter to the CSIR strategy, but more importantly, values that matter to South Africa, because of course, and I'll talk to this later, the key part of our mandate is to improve the lives of South Africans and the continent in general. Because too often we confuse activity and looking busy with achievement. We chase these titles, awards, committee seats, or the appearance of being busy, but none of those on their own build a real legacy. So in my view and in my experience, the real success has come by the knowledge we've generated, and very importantly, the capacity you build, because you can't grow and climb alone. As you climb, remember that if you're leaving those that work with you and are surrounding you behind, you're actually probably defining success in the wrong way. And this is where our CSR strategy is quite clear. The mandate for us to do science and technology that strengthens South African industry and improve people's lives means that our projects, partnerships, and innovations should always connect back to the real-world outcomes. And so, for me, enabling industries to grow, to compete, to innovate, making real change and impact in society is what success really, truly is about. And I'll touch a little bit about the evolution of the career ladder. It's a painful word for some of us, but recently the emergence of the new RDL, in actual facts, aligns very, very well to this, because it gives you many more opportunities and many more paths to define your success as you grow at the CSIR. And it really, truly reflects the shift. We've heard about the sign-up strategy. Sign-up was a program to move to the industrial support strategy, but the new RDL then aligns to this and reflects this very shift. It's no longer about how many papers you've published only or how many committees you've sat on. It's also about how your work aligns with this mission to translate research into impact and building industrial partnerships and solving these very big national challenges that can create impact in society. So I mean, I guess it might sound like redefining success is a high-level thing that sits somewhere far. It's a philosophical question and advice from me. It's actually quite practical, because if you, first of all, choose what to care about, as I alluded to in the first idea, and you redefine what success means for you in alignment to this big mandate that we pursue, you actually will find growth, particularly with this new evolving RDL ladder. When you stop giving Fs about these superficial metrics and start giving them to real impact, you will truly see your career accelerate, and that's my experience, and that's how, from candidate to chief, I found the path. So the last lesson, and this one is a bit brief, is that, I mean, failure is part of the game. And every one of us faces failure in all parts of life. Certainly at work, we face a lot of failure. We know about the elusive reviewer number 2. When you submit publications, reviewer number 2 is always ridiculous. And they may find your work interesting, but not significant. We suffer many proposal rejections. Fundraising at the CSIR is part of what we do and what we must do to be sustainable. And you have to have a thick skin, and you must be able to be resilient to heartbreak, because the vast majority of submissions we make will be rejected. And the nice thing is that it's okay to fail. The TD panel may reject your TDs quite often. Huge projects even come to a halt and stall, but that's part of what we do. In actual fact, I believe that we can confidently say research, development, innovation is actually driven by failure, because what might appear as failure and what might be received as failure is just feedback. It's just data. It's quite interesting that unexpected results, or results that may seem unwanted, are actually results that then redirect and guide what to do next, and actually result in you reaching that outcome that you might have wanted. So if you only care about never failing, you'll never try anything bold, and only pursuing safe science, overly safe science, I think it will rarely change the world, and we will rarely reach the impact that we're actually pursuing as CSIR scientists. So think about the breakthroughs we celebrate: new technologies, great green innovations, partnerships with government and industry. All of them were built on countless failures. What mattered wasn't avoiding this failure, but learning from it, persisting through it, and keeping sight of the mission. So, as I've said, we've had many, many failures. We have freezer breakdowns, we have rejections, and then sometimes even betrayals by your colleagues and partners. It's all part of learning, and the joy of reaching these big breakthroughs, the joy of mentoring brilliant young scientists like yourselves and seeing our work and its place makes all of the failure quite worthwhile. And that's the third and last lesson. I've taken a very long and very inspirational book and tried to break it down into just these 3 lessons. And so as you embark on your own journeys as emerging researchers, the bottom line and the advice I wanted to share was that don't give your energy to distractions, don't measure yourself by the wrong yardsticks, and don't be afraid to fail. Rather, fail loudly, bravely, and with purpose, and you will actually reach the goal of growth and making impact as a CSIR emerging researcher. So give your Fs, and I won't say that word again. Give your Fs carefully and selectively to the science, partnerships, and the problems that, even when solved, will make a real difference in people's lives. So colleagues, with these few words, I welcome you to the Emerging Researcher Symposium 2025. I know there's a theme, and the theme was, at least in the guiding notes I received, was written out as 80 years of innovation, showcasing the next generation of research, development, and innovation. And so while the CSIR celebrates 80 years of groundbreaking work, the next 80 years are shaped by all of us in this room, the emerging researchers of the CSIR that we would like to see. Surely in less than 17 years, I took too long. In 5-10 years at the helm and leading this wonderful organization towards innovation. And the key thing is that you, as the next cohort of innovators, you must choose wisely where you focus your time, your passion, and your brilliance. So I wish you well in this symposium, and I wish you well in your session. Some of you expressed a sense of nervousness as I was having a chat it's no big deal. We are all friends, and I think that the Emerging Researcher Symposium is a wonderful training grounds where you hone your skills in presentation in a wonderful, high-profile venue, but with friends surrounding you. So I wish you all the best with your sessions, and if there are prizes, I don't know, if there are prizes at the end, I wish you well in getting that nice outcome. Thank you very much.
You see now, we've got different problems. Dr. Tsekwa took my step. Now I don't know if you guys can see me. No, it's fine. Don't worry. Yeah, if I'm audible, it's okay. But really, thank you for sharing the wisdom that you've shared with us, both on your reflections, going from an emerging researcher yourself in the CSIR to where you've progressed as a chief researcher, which is where I think a lot of us would like to see ourselves as we progress through the CSIR and work on all of the exciting research that we're doing in our different areas. In isiZulu, they say indlela ibuzwa kwabaphambili, which loosely translated means that we learn from the wisdom of those that have gone before us. And I think you really shared some pearls of wisdom with everybody that's in the room today, including from that book, which has a word in the title that I will not repeat. And so really thank you for the practical wisdom that you've shared, and I hope all of us will carry the words that you've shared with us long after the symposium, even as we may get frustrated by that career ladder, that we'll stick to stick to the course. There's three things that I also just wanted to share before we get into the first session. Just to let you know that we are streaming the session on CSIR New Media, which is on YouTube. There is also a QR code by the registration desk. If you can, please, during the breaks, just make sure that you're scanning that QR code so that we are able to capture everybody that was in attendance today. And then the third announcement that I just wanted to make was a reminder. I'm sure you are aware, but the CEO, Dr. Dlamini, will be with us tomorrow morning to just share some insights. So I really would encourage that we are on time for tomorrow's sessions. We did start a little bit late today, but if we can, please just try to be on time tomorrow for the CEO's remarks. So that leaves me now with handing over to Dr. Nobuhle Majozi for the first session on health and biomedical, which is to run until around 10:15. I really do hope that we all engage with the presentations that we'll be receiving, and good luck to all the speakers. Like Dr. Tsekwa said, no need to be nervous. You're an expert in your field, and we're all really excited to hear about the work that you've been doing. Thank you.
Thank you, Twanelo. Yes, my name is Nobuhle Majozi. I'll be chairing this session this morning, the health and biomedical. So first things first, the presenter has 20 minutes to present, but then we request that you do 15 minutes, and then the last 5 minutes will be for questions and answers. Okay, our first presenter is Lerato Mabe. She is a postdoctoral researcher with the Advanced Food and Agriculture in the CSIR. Her presentation is high sensitivity nested PCR for enhanced diagnosis of bovine brucellosis. Welcome.
For next slide.
Okay. Thank you.
Thanks. I definitely need that one.
Better? Good morning, everyone.
I am Lerato Mabe. I feel like I need to sing just to shake the nerves off a little bit, okay? As Dr. Majosi has said, I am a postdoctoral research fellow in the aquaculture and animal health group within the Advanced Agri and Food Cluster. Today I'm just going to talk about some of the work that we've done developing a high sensitivity nested PCR to enhance the diagnosis of bovine brucellosis. This work formed part of my PhD, which I did under the supervision of Dr. Isa Suleiman and Prof. Oriel Tekiso. Bovine brucellosis is an important but often underestimated disease in South Africa, and our aim was to develop a diagnostic tool that is more reliable, that is faster, and that is safer to use than what's currently available. Okay. So bovine brucellosis is a bacterial disease that is caused by Brucella abortus, particularly in cattle. It significantly impacts both the health of animals as well as the productivity of small-scale and large-scale farming operations in South Africa and in many other countries. It is also a zoonotic disease. This means that it can be transmitted from animals, or cattle in this case, to humans. And this poses a significant risk to farmers who work directly with these animals, veterinary doctors, laboratory technicians, as well as anyone who comes into contact with infected animals or works with infected animal carcasses. With a global incidence of approximately 500,000 cases per year, unfortunately in South Africa, the incidence of human brucellosis is currently unknown because there's currently no active human brucellosis surveillance program within the country. And because this pathogen can infect multiple organs or any organ system in the body, clinically diagnosing it is very difficult because it produces symptoms that are similar to many other diseases. So this is a serious problem when it comes to human health, because if left untreated, the disease can cause chronic debilitating illness that has long-lasting effects. Unfortunately, available diagnostic techniques have a lot of challenges. For instance, bacterial culture, which is currently the gold standard test for diagnosing brucellosis, is very time consuming. It takes a long time, approximately, or it takes about weeks to months for the diagnosis to be complete. And also, it exposes laboratory personnel to dangerous pathogens as they have to work with live bacteria. While serological tests are considered relatively safer and faster than culture, they lack sensitivity, which means that positive cases of brucellosis can be missed while using serological tests. But also, they can cross-react with pathogens that produce similar antigens as Brucella, so thereby leading to false positives. And that is also an outcome that we do not desire. In South Africa, the diagnosis of bovine brucellosis currently is based on a combination of these two methods. But because these methods are either slow or lack sensitivity, it means that outbreaks often go undetected, allowing the disease to spread uncontrolled and posing or leading to significant economic losses in the livestock sector, as well as posing ongoing health risks to humanity. So this brings us to the heart of our work, where we then turned to molecular diagnostics in order to mitigate some of these challenges. And our aim was to improve the time it takes to get a brucellosis diagnosis, as well as to improve the reliability of the assays. So what we did was to develop a nested PCR assay and test this nested PCR assay directly and compare it directly with an existing PCR protocol. Our work included obtaining milk and serum samples from cattle across different provinces in South Africa and rigorously testing both methods side by side. This gave us a clearer picture of how our assay performs in the real world. Okay, so here what you see is just a side-by-side comparison of the performance differences between the published PCR protocol, which I will refer to as the reference PCR from now on, and our nested PCR. This is just one of many examples, but I couldn't really show everything for this presentation. So what you are seeing here, shown by the arrows, is, can I point with this thing? Okay. So what you are seeing on this side, right, is this right, my right, left? Yeah. Yeah. So what you're seeing on this side is the performance of our nested PCR, and on this side is the reference PCR. So what you can see is that the nested PCR was able to detect Brucella DNA even where the reference PCR failed. This is just another example showing you our nested PCR picking up Brucella DNA where the reference PCR couldn't. So this just shows you how efficient the nested PCR was against the reference PCR. And what we also noticed was that the nested PCR was much more, it was not just sensitive in picking up infections, but it was much more robust than the reference PCR, as it was able to resist inhibitors of PCR, as well as it showed resilience to low DNA yield and low DNA purity. And that's the reason why with the nested PCR, we didn't get a lot of the non-specific banding patterns that you see with the reference PCR. When we compared the sensitivities of the assays, the reference PCR had a very low diagnostic sensitivity of about 3%, whereas the nested PCR boasted a 100% sensitivity. And when we compared the performance of the nested PCR in milk and in serum samples, we found that the assay performed consistently across sample types, highlighting it performed consistently across sample types, highlighting its ability to perform across various sample types. So what you can see here is that the nested PCR actually picked up more Brucella in both milk and serum samples, whereas the reference PCR failed to pick up some of those infections. And this just highlights the value of the nested PCR in confirming suspected cases of brucellosis. Okay, so while this was exciting news for us to see our assay performing so brilliantly, it also was a cause for concern because a lot of the samples that we had received and tested with our nested PCR had been reported to have tested negative for brucellosis by our sample donors. So this just suggests that the incidence of brucellosis in South African cattle could very well be higher than anticipated. And this just highlights the existent diagnostic gap that critically needs to be addressed, which is the reason why we decided to develop this nested PCR. Okay, so while that was exciting to see these good results, before we got ahead of ourselves, we decided, let's just make sure that what our assay is picking up is actually Brucella abortus. So we took some of the nested PCR products that came back positive for brucellosis, and we sequenced them, and then we blasted them against an entire sequence database on GenBank. Now, this sequence database will have the sequences and genomes of anything you can think of that's there on Earth: human beings, microorganisms, macroorganisms, everything that you can think of. So when we blasted our sequences against the database, we found, and this was the raw sequences, we found a 97% nucleotide match exclusive to Brucella abortus sequences in the database. And then what we then did was to run a phylogenetic analysis, and that analysis showed us that our sequences, which are depicted by the letters and numbers, they clustered with known strains of Brucella abortus. This gave us a strong confirmation that what we were picking up was actually Brucella abortus, and that our assay is actually accurate and specific. Okay. So having done that, we then decided to continue and validate the assay on all the samples that we had received, totaling 441, which included milk and serum samples. And then from the results, we can successfully say that we have developed successfully and validated a nested PCR that can detect Brucella abortus, specifically Brucella abortus. And we are also happy to report that this assay has now reached a technology readiness level 6, a technology readiness level 6, which means that the assay is now ready for deployment or further scale up. And we are also excited to announce that this technology has been accepted at TRL level 6 as a technology demonstrator by the CSIR TD panel. So in conclusion, the work that we've done goes beyond just developing a more sensitive diagnostic assay. By developing an assay or an assay that is improved in sensitivity, we are not just advancing science. We are protecting livestock, we are securing livelihoods, and we are safeguarding human health across South Africa. And I just want to leave it there for now. Thank you.
Thank you, Lerato, for that very, very interesting and critical presentation and research. Now we're going to take questions.
Hi, Lerato, and thank you for the very interesting talk. So at the beginning of your talk, you indicated the lack of the surveillance systems in South Africa. That's what motivated you to go towards the nested PCR method. So my question is, now going beyond maybe South Africa, maybe looking in Africa or even worldwide, what was available and couldn't we use that and then calibrate it to our unique maybe conditions? Yeah.
Okay, thank you for the question. We did look at what was available in other parts of the world, and what we found was that many of the assays that are currently available, that have been developed for Brucella abortus, are not actually specific to Brucella abortus, because many of the sequencing technologies that came out a bit later on in the years. And because of that, Brucella sequences were not readily available to many scientists. So the assays that were developed prior to the coming out of the whole genome sequences of Brucella were developed thinking that these assays would be specific to brucellosis. But with the advent of whole genome sequencing and people having access to entire genomes, we were able to see that actually the assays that are currently available are not specific. So they can pick up brucellosis, but they can also, caused by Brucella abortus, but they can also pick up Brucella melitensis, Brucella ovis, Brucella suis. So we then developed a nested PCR that was specific.
So is this Brucella's strain a South African issue, or is it shared by other countries, or is it specific to us?
Brucella abortus affects cattle in South Africa, but it does also affect cattle in other parts of the world. Yes, thanks.
Okay, next question.
Hi, thank you for the great presentation. You mentioned that traditional methods of detecting Brucella could take weeks up to a month sometimes. So with your current pipeline, how long is the detection time? And also, are there any strategies that maybe you've developed for on-field or within the farm detection of this pathogen? Thanks.
Thank you. Okay. With collecting a field sample, extracting DNA, and testing that extracted DNA with our nested PCR, it could take a day to get a result. But that would depend on how long it takes also to get a sample from the field into the lab. DNA extraction takes maybe an hour, 30 minutes. The nested PCR takes about 2 hours. So within a day, if you can get your samples into the lab, you could have your results. In terms of whether we've looked into, I'm not sure if you meant we looked specifically us. Okay, so we did develop another technology. It's called LAMP, Loop Mediated Isothermal Amplification. And I don't want to talk too much about that IP. I don't want to get into trouble, but we did develop that technology, and we are trying to get it to be adaptable to the field. Yes, thanks.
Thank you for those interesting responses. Another question? We're left with 2 minutes of questions. Prof?
Thanks, Lerato. It was a good presentation, but I'm still not, having listened to you, which is a good presentation, I still don't understand what is NPCR. Does PCR stand for something?
Oh, thanks, Prof. So NPCR stands for nested PCR, So PCR stands for polymerase chain reaction. I'm not sure if you are familiar with polymerase chain reaction, but this is basically like a method of amplifying DNA or multiplying copies of DNA to make more of it so that you can be able to detect whatever pathogen or whatever you're trying to find within a sample, a small sample. Yes. So the nested PCR is just a derivative of the PCR, which works, so PCR works using a set of primers, and then the nested PCR works using two sets of primers. So where PCR will, the primers of the PCR will attach to one strand of a forward strand of the double-stranded DNA, and then the reverse primer will attach to the reverse strand. These will have been separated in the reaction. Then they will, the primers will then extend the strands and form a separate double-stranded DNA. And then when that happens, the nested PCR then comes and amplifies within that product. So it makes the whole reaction even more specific. I don't know if I explained it well.
Okay, another question?
Hi, nice presentation. I'm just curious as to whether the PCR assay that you use, the reference one, is that like the industrial standard, industrial benchmark? Because like I saw, you said its sensitivity is like 3%, which like I assume low. So I'm wondering, is it the industrial benchmark that you're supposed to be comparing to? And if not, what is the industrial benchmark and how does your stuff compare to the industrial benchmark?
Okay, thank you. So the PCR that we used is not the industrial standard, right? What we wanted to do was to compare our assay to an assay that targets the same region that it was designed on. So what we did was to take an assay that was designed based on the same target region that we used to see how the two would compare. Remember, I mentioned that the technology is currently at TRL 6. To move it up the TRL level, we will still need to do more validations. So we will then compare the assay to the industry standard.
I'll ask a question. Is this disease region-specific within South Africa? And the samples that you used, were they across the whole country or certain areas?
Okay, thanks, Doc. When you read, literature will tell you that it's region-specific, but we obtained samples from all 9 provinces, and all 9 provinces tested positive. Yes. Thanks.
Let's give it a clap. Okay, we move on to our second presenter, Rachel Brown. She is a master's student, and her presentation is generating brain organoids from mouse embryonic stem cells as a model of early neurodevelopment. Rachel, welcome.
Good morning, everyone. My name is Rachel Brown, and I'm a second-year master's student at the University of Cape Town. And this morning, I just wanted to give you a little bit of an overview of the data that I've collected during my project, which was titled Generating Brain Organoids from Mouse Embryonic Stem Cells as a Model of Early Neurodevelopment. So just a little bit of background into brain development in vivo. So brain development is a very complex process. It's quite protracted. It begins in the 3rd gestational week of development, and it continues all the way until about the mid to late 20s. What we see is the emergence of a range of different cell types from a seemingly uniform neuroepithelial cell layer. So what happens with these neuroepithelial cells is they form your radial glial cells. These radial glial cells then can undergo one of 2 processes. They either undergo gliogenesis to form your non-neuronal cell types, like your oligodendrocytes and your astrocytes, or they undergo a process called neurogenesis, which is the formation of your neurons. How this takes place is either directly through asymmetric division, otherwise indirectly through the formation of these intermediate progenitor cell types. And what you'll see throughout this course of development is an increase in the diversity of the cell types of the developing cortex. And you'll see over this time period, the areal identity of the brain is specified. So we start with a more simple structure early on in development, which then forms your more complex dynamic structure of your fully formed brain. And we see that this is a very, as I said, complex process and requires the coordination of many genetic processes. And disruptions in these processes can lead to individuals being at an increased risk of developing a neurological disease. What we see when we look in vitro is the starting point of your pluripotent stem cell line. The first structures that form are these neural progenitor rosette structures. And what these mimic are the early formation of the neural tube, where the neural tube closes to form your central canal of your central nervous system. This then further develops to form a more complex brain organoid structure, which I'll give you a little bit more information about now. And there's still some questions being asked as to how closely this mirrors our brain structure that we have once it's fully formed. But I'm going to give you a little bit more information on how we study the brain in vitro. So as you can see here, there are a range of different models that are used to study brain development, with your ideal model being your human brain tissue, either your post-mortem tissue or tissue that is resected during surgery. As you can see on the left-hand side here, we've got an increase in complexity as we move from our 2D primary neuronal cell culture models all the way to our ideal model, which is our human brain tissue. What you can also see on the right-hand side is an increase in limitation as you move from your ideal model down to your 2D neuronal cell culture model. So in the case of your human brain tissue, we see that there are some limitations associated with its low accessibility and also low throughput. It's really difficult to get access to this brain tissue, and it's also very challenging to keep this tissue alive for studies of your brain development. And again, you would only get tissue from a specific time point. You wouldn't be able to see that develop. The next more ideal model would be your brain organoids. Now, these brain organoids, I'll get into a little bit more detail about them now, but they formed from your pluripotent stem cells in culture. And you see, although they have many applications, there are still some limitations related to its limited maturation and then the organoid to organoid variability. If we look at our animal models, I'm sure a lot of you know what these entail. Obviously, there's lots of species-specific differences if you're studying human brain development using a mouse model, for example. Also, lots of ethical considerations with that. Looking at your animal and human tissue slices, the major limitation with this is the fact that it's a 2D model. Although you do have intact tissue, you don't have that same cell-to-cell and cell-to-ECM interaction that you would see in a 3D model. And then lastly, with your 2D primary neuronal cell culture, again, a 2D, that's a limitation there, and then also you don't have the surrounding supporting cell types. So you can see that there is a need for a high throughput, reliable model that we can use to study brain development. And so I'm going to give you a little bit of an overview into brain organoids, but before we do that, I'm going to give you just a brief introduction into stem cells. So there are 2 different types of stem cells. We either have our iPSCs, so our induced pluripotent stem cells, or our human embryonic stem cells. Now, although these 2 cell types have very similar properties in terms of their pluripotency, they are different. So in the case, in the case of your human-induced pluripotent stem cells, these cells are generated from your adult somatic cells, such as your skin fibroblasts. These are taken from a skin biopsy, which is then broken down and minced into your skin fibroblast cells. What happens now is you then have the overexpression of 4 transcription factors: OCT4, c-MYC, KLF4, and SOX2. And the overexpression of these 4 transcription factors allows these skin fibroblasts to revert back into a pluripotent state, namely induced pluripotent stem cells. In the case of your human embryonic stem cells, these cells are extracted from the inner cell mass of the blastocyst. So the blastocyst is the very early stage embryo, and you take these cells from the inner cell mass, you extract them and culture them. Although these models are pluripotent, which means that they can be differentiated into cells of all 3 germ layers, they do have a limitation in the fact that they are still 2D. And so this is where our 3D models of stem cells come into play. So the first 3D model of stem cells to form in culture are EBs, or embryoid bodies. And these are aggregates of your pluripotent stem cells which are formed in suspension culture. Due to the pluripotent nature of these cells, they can then be differentiated into cells of any type. Of interest in our lab are these cerebral organoids. So cerebral organoids are aggregates of neurons and glia formed in suspension culture, and they have been shown to be the ideal model to study brain development in. They have the ability to demonstrate the cell-to-cell interactions and the cell-to-ECM interactions of the brain, and also they have intact neuronal networks, which is great for your electrophysiology studies and things like that. So, how do we make these brain organoids? How this starts off is we start off with our 2D cells. In the case of this project, we use mouse embryonic stem cells. These cells are then cultured and aggregated in 96 well plates at 2,000 cells per well. The cells are then cultured in differentiation medium for the first 5 days. This differentiation medium contains ROCK inhibitor and beta-FGF for the first 3 days. ROCK inhibitor is important for promoting the survival of the cells when they're in single cell state while they're still aggregating. And then beta-FGF is also important for the formation of that initial embryoid body structure. On day 3, medium is changed and those 2 supplements are removed. And then on day 5, the aggregates are transferred into neural induction medium, which contains N2 supplement and heparin, which are both essential reagents for the differentiation of your pluripotent cells into your neural progenitor cells or your neural stem cells. We then collected the aggregates on day 11. We are still in the process of optimizing day 11 through to 18 of culture, but for the sake of this presentation, I'm just going to present data from the first 11 days. So this is what our brain organoids look like. So we start off with fairly round structures on day 1, quite light in color, and you see over the first 5 days how they start to solidify, they start to round up, and they get a lot darker in color. Then when we move our aggregates from differentiation medium into our neural induction medium on day 6, you see the formation, it's quite difficult to see on the screen, but you see the formation of these lighter buds. So what these are neuroepithelial buds. We have our neural epithelial cells or our neural progenitor cells that differentiate and start to migrate out, forming those initial buds, which then fold and turn to form your brain structure. So there's a bud over there, you see one up the top there, it might be easier to see, but you see the formation of these buds as these cells start to migrate out. And then you'll see on day 11, we have quite a different looking structure to what we saw on day 1. So in order to confirm what we were seeing, we wanted to do some image analysis in ImageJ. So what we did was we took images of our aggregates on every day, and we outlined them and calculated the area, perimeter, and ferrite diameter. And what you'll see here, so area, as you know, is the area of the shape, perimeter is just the border, and then your ferrite diameter is the longest point from one side of the aggregate to another. And you'll see a really nice increase in all of these different parameters. Steady increase in area and perimeter, as well as your ferrite diameter, which confirmed to us that our organoids were growing and they were developing. We then had a look at 3 other parameters, roundness, aspect ratio, and solidity. With roundness, this is just measuring how round the organoid is. You'll see a roundness of 1 is a perfectly round shape. So you'll see the organoids round up over the first 5 days, and then a decrease in that roundness as they start to bud out and form those neuroepithelial buds. The same can be said for the aspect ratio. The aspect ratio is measured differently to the roundness. So what this is you have a major and a minor axis of your organoid. These are measured perpendicular to each other, your major obviously being your longest of the 2 and your minor being the shortest. So the aspect ratio is measured as a major over minor axis calculation. And again, you see a rounding up over the first 5 days with an aspect ratio of 1 being a perfect circle, and then the formation of these buds over the subsequent 6 days. Solidity is just showing us how solid the organoid is, and I'm sure you saw from those images, we have a really nice solid shape to begin with, and then we've got the formation of these more translucent neuroepithelial buds over the subsequent 6 days. Next, we wanted to have a look at the expression of these early neural gene markers in our organoids. So we had a look at our, neural progenitor markers, both PAX6 and Nestin. We see a significant increase in both of these markers in our organoids on day 5 compared to our controls. As I mentioned previously, our embryoid bodies are spontaneously formed. They are generally more pluripotent than your brain organoids or neural embryoid bodies, and so you can see we just use those as our control for this PCR experiment. We then looked at our neuron markers, TUB3 and DCX. We see a higher expression of DCX, which is a marker of more mature neurons, which was very promising that we were seeing this 5 days into our culture period. And then what was quite interesting was the expression of REX1 in our organoids, which is actually a pluripotency marker. And I was quite surprised to see this because we are hoping that our cells are differentiating and are not pluripotent. But I suspect that this is due to also the presence of the neural stem cell population of these organoids. As you can imagine, they can't fully differentiate over 5 days, and so there is still that multipotent neural stem cell population there. 4 more markers we had a look at, our midbrain and hindbrain markers. So ISL1 being your midbrain marker, OTX2 being your hindbrain marker. Again, a significant increase in expression of both of these gene markers. Astrocytes, again, significant increase in GFAP, which is a marker of our astrocytes. This is also very promising as astrocytes are important during the developmental process. Forebrain marker, FOXG1, again, significant increase in that. What's quite interesting is compared to ISL1 and OTX2, we do see a higher expression of FOXG1, which is suggesting that our organoids are taking on a more forebrain identity. So just in conclusion, our day 5 NEBs show a significant increased expression of all these neural marker genes compared to our controls. Lastly, we did some immunocytochemistry experiments. Firstly, we looked at NUN and F-actin. So NUN is it's a marker of neurons, your early neurons, and we see here a really nice expression of NUN in all of our cells. So we see this adoption of a complete neural fate. We then saw the expression of F-actin in these little luminal shapes over here. And F-actin is a marker of the cytoskeleton. So this is showing us the formation of these neural rosette-like structures that I spoke about previously, where you have this really nice little lumen forming, which is surrounded by your neural cells. We then had a look at SOX2 and TUJ1 expression, and we see, so SOX2 is a neural progenitor marker, and we see the really nice formation of these, again, these neuroepithelial buds, where our neural progenitor cells are migrating out and forming this bud, which is what we suspected from our morphology analysis. TUJ1 is a marker of both your mature and your immature neurons. You see really nice localization of TUJ1 in the outer cells. What we expected to see was these neural projections forming. However, we do know that TUJ1 is associated with nuclear cellular processes, and so that's why we see it here. We're hoping that at later stages we do see those projections forming. So just in conclusion, we saw that our MESCs can be successfully differentiated to form our brain organoids with neural rosette-like structures. This was confirmed through a range of different methods: morphological analysis, RTQ-PCR, ICC. We have generated the first cerebral organoids in Africa, which is making the initial step towards creating an African-specific model to study brain development and health. And then, obviously, this work has significant implications for our laboratory and the broader African scientific community. I just want to thank my supervisor, co-supervisor, and all of our funders for their continued support. Thank you for listening.
Okay, thank you so much, Rachel, for such an interesting presentation on the brain. Okay, we'll take questions.
Okay, good morning. My name is Luando from the NGEI cluster. I wanted to actually have two questions. Being a layman in this whole topic, I wanted to understand, or wanted to ask if my understanding of organoids is correct. So you said organoids are a collection of cells that get to form the whole brain at the end of everything, right? Okay, so I wanted to understand if whether, because I've heard of the concept of 3D printing of human organs, and that it stems from having to donate like cells from, if let's say the organ that is to be developed is for you, so they will need to take cells from your existing organ and then they can replicate that. So that does this study of brain organoids also correlate with the idea towards 3D printing of human organs? Can that be something that can be achievable with what you are doing here?
Yes and no. So what we are doing is we are allowing these organoids to spontaneously differentiate. Obviously, we are directing them towards a neural ectodermal lineage, which means that your mesodermal cell types and things like that will not be present in the organoid. So it provides kind of room to co-culture and produce an organoid that then has all of those cell types. With your 3D bioprinting of organoids, that obviously is a little bit more controlled. You've got a very specific amount of cells that are being added of a specific cell type. Obviously, you do need to make sure that you are using cell types from the same individual, that you don't have any of that interaction happening. So in the sense, yes, that is it's still an organoid, but it is a different stream, because here we're working with like spontaneous generation, because we're wanting to see if we provide it with what it needs, what's going to spontaneously happen, and that will inform what happens during early neural development, which we can't obviously see in vivo, which could then inform, okay, at this point, this cell type, so this is when our astrocytes come in, which will then inform how we do the 3D bioprinting. Does that make sense? Okay.
Thank you for a very nice talk. Very interesting. I'm interested in finding out, or rather curious, the endpoint is, of course, to use these as a model for
Sorry, I missed that.
The endpoint is to use these as a model?
Yes.
Yes. So how long are the organoids viable? So if you were to create your organoids and then want to test them, or test a treatment, or whatever your end goal is, how long do they last?
So generally, in the global north, they've managed to culture these organoids for 300 days, so for a very extended period of time. Obviously, that's very resource intensive and requires a lot of work. So generally, while they're still in culture, they are viable. As long as you're feeding them, as long as you're giving them what they need, they they do generally stay viable. But then if you're wanting to do other downstream, so like for example ICC, we obviously fix them, but if you're wanting to do your electrophysiology tests and stuff, you need to make sure that you keep them, because they can die quite quickly when you remove them from their culture medium. So you would have to figure out a way, I haven't done that personally, but you'd have to figure out a way to make sure that they stay within their culture conditions. Yeah.
One last question. Good morning, Rachel.
Morning.
Great presentation. I just have one question. So what are the biggest limitations of using mouse embryonic stem cells derived organoids in comparison to human organoids?
So obviously you've got the major difference, which is the species-specific differences. Our lab, we wrote a review which compared the timing between your human organoids and your brain organoids. And generally what happens is your mouse organoids tend to develop a lot faster than your human organoids. So in terms of that timing, you're going to have it happening a lot quicker. So that is a limitation. Ideally, we are moving in the direction of using human cells to create human organoids, because ultimately that is more appropriate to study human development. But for now, what we're seeing are promising results that's mimicking what happens in the early development of the mouse brain. Yeah.
Okay, let's give our speaker a round of applause.
Okay, the last speaker of the session is Potsiso Koma. And she is a PhD student, and her topic is investigating the antioxidant, mycobacterial and antibiofilm activities of medicinal plants against Mycobacterium smegmatis.
Good morning, everyone. My name is Pujiso, a PhD candidate from the University of Limpopo, and today I'll be taking you through my presentation titled Investigating the Antioxidant, Antimycobacterial and Antibiofilm Activities of Medicinal Plants Against Mycobacterium smegmatis. Tuberculosis is an infectious airborne disease caused by Mycobacterium tuberculosis complex, which primarily affect the lungs. It is resistant to the current available treatment methods, and this is due to it developing new survival mechanisms, bacterial cell-to-cell communication, which promotes biofilm formation and dispersion. The biofilms act as a physical barrier, preventing antibiotic penetration and protecting the bacteria from environmental stressors. The first-line drugs induce side effects, which makes it difficult for patients to continue with the treatment for a minimum period of 6 months. So this led to research to uncover new alternative methods to treat and manage TB using medicinal plants. Traditional medicinal plants have been reported to have bioactive compounds that can be used as alternative sources of therapeutic agents to control and manage TB. The study aimed to evaluate the antioxidant, antimycobacterial, and antibiofilm activities of medicinal plants against Mycobacterium smegmatis. Here are my objectives. The plants were collected at the Lowveld National Botanical Garden in Mpumalanga, and some of the plants were collected within the University of Limpopo. They were then dried at room temperature, ground to fine powder, and 1 gram of the plant leaf material was extracted with solvents of varying polarities. They were then transferred into pre-weighed vials, and the mass of the extracts were calculated. And figure 1 shows the mass extracted by the different solvents, and it was observed that methanol had high extraction yield as compared to all the other extracts. except for seed labrum and Eucophagia capensis, which had their high extraction yield from the water extract. This shows that these plants had more polar compounds because the methanol and water are polar solvents which tend to extract more polar compounds. The extracts were then reconstituted to 10 milligram per mil with acetone, and phytochemical analysis, quantification of phytoconstituents was done using the standard chemical methods. It was observed that Dicrostachys cinerea had the highest total phenolic and tannin content, Hypericum ruperianum with the highest total proanthocyanin content, but Ledger salvifolia with the highest flavonoid content, while Combretum apiculatum had the highest flavonoid content. Since these phytoconstituents have been reported to have antioxidant, antimicrobacterial, and antibiofilm activities, we then looked at the antioxidant activity using the DPPH free radical scavenging activity assay, whereby in this assay, plants with antioxidants transfers a hydrogen atom to the hydrazine, and the nitrogen atom of DPPH is reduced. It forms a purple color when dissolved in methanol, and the presence of antioxidant compounds are shown by the yellow bands against the purple background. We then have to quantify the amount of oxidant that will be able to inhibit the free radicals of the reactive oxygen species. And it was observed that Dicrostachys cinerea had activity comparable to that one of ascorbic acid, which then shows that the antioxidant activity from Dicrostachys cinerea might be from the total phenolics that were quantified in the plant. With the ferric reducing power assay, the plants with antioxidants reduces the ferric ions to ferrous ions, and it forms a brisant blue color. Ascorbic acid had the lowest IC50 value, followed by seed labrum. We then looked at the antimicrobacterial activity using the broth microdilution assay and bioautography assay, whereby INT was used as an indicator. It is based on the dehydrogenase activity of viable cells to convert the INT into a formazan color. The clear bands against the pinkish background indicates where the phytoconstituents within the plant extract were able to inhibit the growth of Mycobacterium smegmatis. And it was observed that Buddlegia salvifolia, Carissima crocapa, Compritum apiculatum, and Buddlegia saligna had promising bands as compared to other plants. We then looked at the minimum inhibitory concentration and minimum bactericidal concentration, whereby the minimum inhibitory concentration is the lowest concentration of the antimicrobial agent that will be able to inhibit the growth of the bacteria. And with the minimum bactericidal concentration, it tells us about the minimum concentration that is needed to kill the bacteria. It was observed that Combretum apiculatum had the lowest minimum inhibitory concentration of 0.16 milligram per mil, which was similar to the minimum inhibitory concentration of the rifampicin. Ooh. Of the rifampicin. And it also had the minimum bactericidal concentration from the same extracts of 0.63 milligram per mil. We then looked at the combination effects to check if whether the positive control and the plant extract can work together to inhibit the growth of Mycobacterium smegmatis. It was observed that a combination between Haphophyllum cafram and rifampicin, Combretum apiculatum and rifampicin had an additive outcome. A combination between dicrostachys cinerea and rifampicin had an antagonistic outcome, while the other combinations had an indifferent outcome. Looking at the antimicrobacterial activity from bioautography and broth microdilution assay, 5 plants were chosen for further analysis, which are Badlegia saligna, Hapaphyllum cafram, Badlegia salvifolia, Compritum apiculatum, and Carissa macrocarpum. The effect on growth phases and inhibition of growth was evaluated using the growth curves at 2 times MIC, MIC, half MIC, and quarter MIC value. It was observed that all the plant extract had good activity against Mycobacterium smegmatis. Since this method does not tell us about the number of viable cells within the extract, we then looked at the enumeration of viable cells using the time kill assay. Only 1 plant, the positive control and the untreated, are shown on the slide. And it was observed that on figure 5A, there was countable number of colonies from time of inoculation until 18 hours, as shown in the graph. And then at 24 hours, there were too many to count colonies. With figure 5B, which was the positive control, the rifampicin had a bacteriostatic effect against the organism for a period of 9 hours. And then after 9 hours, at 12 hours, there were countable colonies until 18 hours. And with the untreated, which is figure 5C, there were too many to count colonies. Mycobacterium is associated with exopolysaccharides, which plays a huge role in motility and biofilm formation. The quantification of exopolysaccharides was done using the phenol sulfuric acid assay. Where in this assay, carbohydrates react with sulfuric acid to produce monosaccharides, which then reacts with phenol to produce a colored complex, and the color intensity is directly proportional to the number of exopolysaccharides present. It was observed that less exopolysaccharides were quantified at MIC value as compared to half MIC value. And Badlegia salvifolia had the lowest number of exopolysaccharides quantified. In motility, these exopolysaccharides Mycobacteria uses a sliding motility to move around. So in motility, these exopolysaccharides play a huge role to prevent the bacteria from friction by the surface area. So it was observed that there was 100% inhibition at MIC and half MIC value when it was treated, when Mycobacterium smegmatis was treated with rifampicin, followed by Hapaphyllum cafram with 82% and 71% at half MIC value, while Compritum apiculatum had the lowest inhibition at half MIC value. In biofilm formation, these exopolysaccharides play a huge role as they contribute to the formation of an extracellular matrix which shield the bacteria from the antibiotics. The prevention of biofilm formation was evaluated using the crystal violet staining assay, and it was observed that there was no significant difference between the ability of rifampicin and Compritum apiculatum to inhibit initial cell attachment at all the concentrations. Low activity and enhancement of activity. Enhancement is the one with the negative inhibition, was observed when the plant extracts were treated with Hapaphyllum cafram, Badlegia salvifolia, Carissa macrocarpa, and Badlegia saligna. Looking at the prevention of biofilm formation, it was observed that all the plant extracts had good activity with an inhibition greater than 50%, except for Buddlegia salvifolia at MIC over 8. We then looked at the eradication of the mature biofilm. And it was observed that there was no significant difference between, the Combretum apiculatum and the orange one, and Buddlegia salvifolia had good inhibition with an inhibition percentage greater than 50%. Low activity and enhancement of activity was observed from the other extracts. In conclusion, the selected medicinal plants exhibit potent antioxidant, antimicrobacterial, and antibiofilm activities, indicating their potential as sources for isolation and characterization of bioactive compounds. Here are my acknowledgements. Thank you.
We'll take questions. The first question.
Thank you for interesting talk. I have to confess that most of it went over my head. So maybe just reducing it to standard grade for me. Please clarify. So in the pictures that you showed where you were showing the bacterial growth, your plates. Yes. So were you saying that maybe the media, was it treated with now your plant extract, and then the bacteria only grew, the growth of bacteria was delayed?
Yes.
So does that mean that in terms of now treatment, that also dictates like how often the patient will have to take the treatment? Okay, thank you.
Hi. Out of interest, I would like to ask, so you obtained secondary metabolites from your plant extracts, right?
Yes.
So I wanted to ask if these secondary metabolites were already existing in your plant extracts, or they were existing from possible prior exposure to the M. smegmatis strain before. So I don't know if I'm making sense.
You mean these ones, right? No, they were existing in the plants.
So it's not from exposure whatsoever?
Exposure might be from the sun, environmental conditions.
Thank you so much for the presentation. I just want to check what informed your choice of plants.
The plants were selected based on literature about the ethnopharmacological use to treat respiratory infections and their related symptoms. Then we looked at the plants that are less investigated, and we chose those ones.
Just a follow-up question from that one. Is there a particular decision that made you choose specifically the leaves other than maybe the stems or the roots?
Based on availability. Because if you take roots, the Environmental Act will not allow you. So you only get less amount. And also in this study, I'll still have to isolate the bioactive compounds. So if I have to use the roots, it means it will be something else. We won't have the plants at some point, yeah.
Hi. Did you purify your extracts out of the acetone?
Did I do it?
Did you purify the acetone out of your extracts?
Come again?
The acetone, you extracted them with acetone? The extracts, you extracted them with acetone?
Yes.
Did you purify the acetone out?
No, because we extracted with the acetone. And then from there, we let the extracts dry so that we can get the mass. Then we reconstitute with acetone because of its feasibility with polar and non-polar compounds. So it allows us to extract more compounds.
You reconstituted with acetone and then treated, or rather plated that?
Yes.
So how can you be sure your bacteria didn't grow because of the extracts rather than the acetone?
Oh, sorry. We have acetone as the negative control.
Okay, cool. Did you do any toxicity testing on the compounds?
I'm still busy with that.
Okay.
Morning. Thanks for a great presentation. You mentioned that if you were to use the roots, you'd have to isolate, right? So, meaning in this case, you didn't isolate, you just took the extract as it is, and then you are working with the whole...
crude extracts, yes, for now. But I'm busy isolating.
Oh, okay. So meaning that you're going to look at which of your bioactives is more active.
Yes.
Oh, okay. Thanks.
Just last one. Just check. Have you published any of this data yet?
Not yet.
There's no publication yet on this stuff. Okay.
Thank you so much for
What's
this.
this?
Let's give our presenters one more round of applause. Yes, our session ends here. Where's our... Okay, next we are proceeding to the poster and exhibition. So we can go and view the posters, ask questions, communicate with the poster presenters. Thank you once again. And we're going to come back at quarter to 11. We'll come back to this hall, quarter to 11.
Give me your love, no holding back, no. We can take it to the stars where you wanna go. Stop, we can't do, don't need to prove you know. Honey, I got what you want, yeah. Under the night sky, I want your eyes all on me. Don't tell me no lies, I always get mine. Don't play with me. So do you want it? Do you want it? Can you feel me? Can you feel me now? Can you handle it? Alright. So do you want it?
It's a difficult process. So, unfortunately, if this type of pathogens happens to and it's not controlled, the transport are susceptible to the it can survive in the soil. What did the self-connect itself connect? So what is the current situation that we have? The spread of this virus. So the current that we have that information, we rely on
where we have our standard, we have this is the mix of standards. We have the cocktail. Yes, the cocktail. The different, like polysaccharides. And then here we have now the reaction in water. We just see what is going to produce every cell phone. And then what do the HPA. And then these single oligosaccharides are quite interesting. People they're prebiotics, so they can be used as antioxidants as well.
part of it is looking at the grade to say how much of whatever minerals we're going to get out of the rock. And another thing it's knowing the structures. Because that goes in, so if you can still mine it, you still have other mines that are no longer mined because of certain issues like for water, different structures. So after the proper process, you have to sit down and look at how to best mine it, the structure of the rock, how you can support it, actually have a mine it. And another thing you have to do is mechanize to allow it to be because they conventional, they
Are we talking to each other? Or to you?
Is he going to be audible here? Good morning to you. Welcome to the CSRC Emerging Researcher Symposium. Can you just try to tell the public what this event is all about?
Yes, definitely. I think this event is the 8th ERS that the CSIR has hosted. It's 80 years of showcasing new researchers, new innovations. It's very exciting to see what the researchers within the CSIR are doing and how they're impacting South Africa and the broader community.
So, what will you say if maybe it's tried to emerge it into inviting other institutions just to try and collaborate to show what other youth are doing out there?
Yeah, I think it's very important. This is really an awesome function to attend and or symposium to attend. It's nice to see what other people are doing, and it actually fosters collaboration because you can see what other people in other areas are working on, and that can actually motivate you to work together on certain projects or help each other with certain solutions.
What message do you have for the young researchers at the CSR who didn't join this year? Maybe a call for them for excitement and interest next year?
They definitely must attend. They're missing out. This is really amazing to see what other young researchers are doing, very passionate about what they're doing, how you can grow in your career. It's very amazing, and yeah, they should come, they should present on their work. Anyone is welcome, any topic is welcome, so they should definitely sign up next year.
What message do you have, an epic message to everyone as we celebrate our values of excellence?
I think if you are excited about what you do, you will definitely live out the epic values. You will be interested in collaborating with different people. You will be invested in your work. You will be proud of what you're doing. And I mean, making an impact in the broader society, that's a very good value to strive towards. And then using that to grow in your career, I mean, that's a win-win situation.
Oh, no, thank you so much. And we are really glad to host you and to have you today. And we are looking forward to today and tomorrow.
Yes.
Thank you so much.
Thank you so much.
Yeah.
You are good at this, man.
to just give our fellow colleagues maybe an additional minute to just make their way to the venue. Then we'll start at 10:50. Thank you. Ngifuna ukubona ukuthi i-helium kwenzakalani ngayo. Okay, it is 10:50, so I'm just going to give the colleagues 10 seconds to just make their way to their seats, then we can proceed. I hope everyone enjoyed the poster session. There's also posters way at the back. It was so sad. The people who were standing that side were very lonely. So please, during the poster session, please also just move across and see them as well. Okay, so we will officially start. So good morning, colleagues, and welcome to the second session of the CSIR Emerging Research Symposium. And my name is Sindisiwe Buthelezi. I will be on health. Continues to drive innovation towards diagnostics, therapeutics, and public health. So this session we will be hearing from 5 promising researchers and who are playing an active role in shaping the future of our health in South Africa and beyond. So before we start, I would like to just do a few housekeeping notices. So number one, I've been tasked to just remind everyone to please register on the actual system as they are collecting the statistics and the data. So not just the manual registration, but please also find someone, think they're at the front desk, to help you enter your details in the app as well. And then please ensure that your devices are on silent, and the speakers will all get 15 minutes to give their presentation. Then there'll be 5 minutes for Q&A. And then to the speakers, when you have 5 minutes left, I'll just show my paper of 5 minutes. Then when you have 1 minute left to wrap up, I'll just stand on the side. All right, so please help me welcome our first presenter, who is Sesianda Masego. The title of her talk is Whole Genome Characterization of Neuroviruses Detected in Children Under 5 years from Gauteng, South Africa. Thank you.
Good morning, everyone. My name is Sesianda Masego, and today I will be presenting my preliminary findings for my master's project. Oh, am I short? Yes, so as I said, today I will be presenting my preliminary findings for my master's project. It is well documented that noroviruses are among the major diarrheal causes. But what if other enteric viruses are silently present, contributing to the overall disease burden? To explore this, metagenomic next-generation sequencing of norovirus positive fecal samples was conducted, which was obtained from South African children under 5 years, and this revealed norovirus diversity and co-infections with other enteric viruses. Diarrheal diseases are among the major causes of infectious disease morbidity and the 14th leading cause of mortality across all age groups. In children under 5 years, unfortunately, the impact is even more severe, with it ranking the 3rd leading cause of mortality. Viruses are the common cause of acute gastroenteritis, including rotavirus, norovirus, sapo, and astroviruses and adenoviruses. Among these, noroviruses have been recognized as the leading cause of acute gastroenteritis across all age groups. And this has been demonstrated by its significant global disease burden, with an estimated 685 million infections reported annually. Children under 5 years are particularly vulnerable, with over 200 million infections and 95,000 deaths reported annually. In sub-Saharan Africa, norovirus accounts for 19% of global infections, with South Africa having a prevalence of between 20 and 30%. We now know that noroviruses exert a significant disease burden, but what exactly are noroviruses? Noroviruses are single-stranded RNA viruses. They are classified within the Calliciviridae family. They have a non-enveloped structure with the VP1 major capsid protein forming an external shell, encapsulating a genome of approximately 7.7 kilobase pairs, which consists of 3 open reading frames. They are a genetically diverse group of viruses divided into 10 genogroups, which are further subdivided into 49 genotypes and 60 polymerase types. Although noroviruses exhibit a significant diversity, human infections are mainly caused by genogroups 1, genogroups 2, 4, 8, and 9. Despite norovirus' significant disease impact, there are currently no vaccines or treatments for noroviruses, with challenges including high genetic diversity and the lack of cross-protective immunity. A key approach in advancing norovirus surveillance is the genomic surveillance, which enables strain tracking and thus informs vaccine design. Although partial sequencing methods have provided valuable information, they lack the resolution required to capture full viral diversity. Whole genome sequencing via next-generation sequencing, on the other hand, offers high resolution and thus detailed genetic insights. Moreover, metagenomic next-generation sequencing approaches, which use non-specific primers, provide an even broader advantage as they allow for the detection of novel and co-infecting viruses, which would have been otherwise missed by using amplicon sequencing. Despite the advantages that these studies offer, such studies are scarce in Africa, even though the continent bears a significant disease impact. And to then address this gap, this study aimed to characterize norovirus genetic diversity using whole genome sequencing and to detect co-infections with multiple norovirus strains and co-infections with other enteric viruses using MNGS. This study forms part of a larger study, the CSIR. Ethical approval was obtained from the UFS-HS-REC and UFS-EB-REC. For this study, a total of 84 archived norovirus-positive fecal samples from children under 5 years old from the Gauteng province, South Africa, between 2008 and 2019 were retrieved from the NICD. To then process these samples, the NETO-VIR protocol was then followed, where viral enrichment was firstly conducted following the necessary steps, followed by nucleic acid extraction using the respective kit, then cDNA synthesis and whole transcriptome amplification, followed by library preparations, and lastly, next generation sequencing using the NextSeq 2000. The Illumina raw reads were then processed using two web-based bioinformatics tools, and those were Genome Detective and Trans-Zuckerberg ID, where low-quality reads were filtered, adapters trimmed, and reads assembled into context, as well as viral sequences identified. The human calicivirus typing tool, as well as the norovirus typing tool, was then used to assign norovirus genogroups, genotypes, as well as polymerase groups and types. To give you an overview of the metagenomic data, 84 norovirus positive fecal samples were processed, of which 55 norovirus full genomes were generated and 2 near full genomes, and with the remaining 27 samples either producing partial sequences or having a few reads to proceed with further analysis. Over 12 million mapped reads were generated, with 91% of those belonging to noroviruses and 9% to other co-infecting viruses. To give you a perspective of my preliminary findings, we will firstly focus on noroviruses. This pie chart depicts the distribution of norovirus genotypes, and evidently G24 was the most predominantly detected strain, followed by G26 as well, and G23 and other norovirus genotypes circulating at lower levels. This bar graph depicts the yearly distribution of norovirus G24 variants, and evidently it indicates that G24 was not a single static strain, but rather several G24 variants, namely Allegheny, Appledoorn, New Orleans, Osaka, and Sydney, appeared and replaced each other over the years. A very interesting observation was the detection of the G24 Sydney variant in 2009 and in 2010, which was way before the major 2012 Sydney outbreaks. This study also observed a recombination frequency of 73.7% and a recombination frequency of 73.7%, and this was indicated by mismatching in the capsid and the polymerase, and this assisted in giving an initial insight into which recombinant strains would have been circulating during this period. Additionally, 9.5 recombinants were collected in the same year and same site. For instance, in 2011, 2 G26P7 strains were collected from site 2, and in 2017, 2 G24 Allegheny P31 strains were collected from site 10. Still on noroviruses, this study observed a co-infection frequency of 21%, with samples containing up to 3 different norovirus strains. For instance, patients with sample number 8215 was co-infected with a G2 strain, a G4 strain, and a G1 strain, G4 strain, and a G24 strain. Now that we've looked at noroviruses in detail, let's now shift our focus to co-infections with enteric viruses, so other enteric viruses. Evidently, rotaviruses were the most commonly detected, which co-infected 35 out of 57 patients, followed by adenoviruses, then Astra, and lastly Sapo. So the darker shade of red indicates a high viral read count, and evidently rotaviruses had a higher viral read count compared to the other viruses. Beyond co-infections with common enteric viruses, the study also detected co-infections with other enteric viruses, and evidently enteroviruses and pericoviruses were the most frequently detected, with other viruses detected at a lower frequency.
And as with the previous heat map, the darker shade of red indicates higher viral read count, and enteroviruses and pericoviruses accounted for most reads. Evidently, metagenomics enabled us to recover norovirus genomes as well as to detect other enteric viruses. And the partial, or the 65.5 recovery of whole genomes was mainly due to there being a few viral reads. And this basically highlights that in as much as metagenomics enables us to capture other enteric viruses, these may be more abundantly captured than the intended target. Despite the partial recovery rates, the recovered genomes did reveal norovirus genetic diversity, with G24 being the most dominant, followed by G23 and G26. So the G24 dominance could be explained by the emergence of multiple G24 strains which could have escaped acquired immunity. An interesting observation, as I have mentioned, was the detection of the G24 Sydney variant in 2019 and in 2010, which was before the major 2012 Sydney outbreaks. And this basically highlights the importance of doing early genomic surveillance, as this will enable us to detect these strains before they become more widespread and thus help us to prepare for future outbreaks accordingly. The high recombination frequency that was observed in the study reinforces what has been widely reported, as it has been reported as a major driving force of norovirus diversity. The 9.5% recombinants that were collected in the same year and site could imply that they're from a shared source. However, transmission chains have to be done to basically determine or indicate whether these infections were indeed linked or independent. Building onto norovirus diversity, the fact that 21 patients were infected with more than one norovirus strain basically indicates the genetic diversity that can occur in just a single infection. And this is particularly concerning, as studies have reported that norovirus co-infections facilitate recombination and thus the emergence of novel variants. The high occurrence of co-infections with enteric viruses could be explained by poor sanitation and unsafe water in the setting, as well as the fact that these pathogens share a fecal-oral route transmission, and thus increased susceptibility may occur after the primary infection. Rotaviruses being the most commonly detected is consistent with global studies, and this is explained by rotaviruses being the leading cause of age in children under 5 years. The fact that high occurrence of co-infections, single enteric infections cause significant morbidity. The fact that high occurrence co-infections were detected in this study and multiple pathogens may infect just a single host, this could lead to increased morbidity, complications, as well as mortality, and this has been explained mostly in children in LMICs. In conclusion, the preliminary findings of this study provide insights for vaccine developments by basically demonstrating both emerging as well as dominant strains. The predominance of G24, which is consistent with global and local findings, reinforces its ongoing role in norovirus infections, and it makes it an attractive target for vaccine development. However, the genetic variability that was observed may limit strain-specific vaccine efficacy, and thus is a need for a cross-protective vaccine. The high occurrence of co-infections that was observed in the study indicates that other enteric viruses are indeed silently present, which may complicate control efforts and thus make treatments less efficient. This then emphasizes the need for a broad multi-pathogen metagenomic surveillance beyond single virus testing. Now that the preliminary findings for this study are now in place, the next steps are to ensure completion, are to determine recombination breakpoints, phylogenetic analysis, elucidating transmission patterns, and this will assist in building a more comprehensive understanding of noroviruses and thus guide future responses as well as prevention strategies. I would like to acknowledge the following. Thank you. Just stool samples, fecal samples.
How big was this sample or the number of feces that you'd got from the patients? How many? How many patients?
Oh, it was 84. Yes.
From one area or from different provinces?
It was from Gauteng. It was different areas in Gauteng.
Any more questions? Maybe another one from myself while the colleagues are thinking. Is the core infection, the viruses, is there one now that you always expect to be there? And does it somehow interact now with the norovirus? Is there a particular reason why it always seems to go hand in hand or with the norovirus infection?
Sorry, can you please say that again?
So the viruses that are co-infecting, are they like particular viruses that you would expect? So if you see the norovirus, you know that the other virus will be there as well. And if that's the case, is there any maybe interaction between these viruses that will that encourages the co-infection?
Not necessarily. So there's no specific virus that is expected to be always co-infecting together with noroviruses. It usually interchanges. Yeah, so there's no specific one.
Is it region-based or?
I think region would or can play a role, because especially because noroviruses are transmitted by unsafe water, etc. So basically, if a region is to have unsafe water, then there could be a high chance of co-infections occurring in that area.
Thank you. Any more questions? Okay, thank you.
So now I'm not sure. All right. Our second speaker is Oyomide. I hope I'm not mispronouncing your name, Abe, whose work lies in the intersection of medical imaging, artificial intelligence, and diagnostic innovation. So the title of the presentation is Lung Cancer Diagnosis from Computed Tomography Scans Using Deep Learning Algorithms and Marvage Pooling and Random Pixel Swap Data Augmentation Technique. Thank you so much.
Good morning. I'm happy to be here. So I'll be presenting results from my PhD studies. It was supervised by Professor Nyati, Professor Kwande, and Professor Piloy. The title of my presentation is Lung Cancer Diagnosis from Computed Tomography Scans Using Deep Learning Algorithm with Marvage Pooling and Random Pixel Swap Data Augmentation. Here is my presentation outline. Lung cancer is the most common cancer for all genders. It is the highest cancer-related deaths in the world. The average, 5-year survival rate for lung cancer is very low compared to other cancer types, which makes detection of lung cancer to be a critical need. For example, if lung cancer detected early, 55% of patients survive more than 5 years compared to when it's detected late, where we can see about less than 5%, who survive. In South Africa, about 8,000 lung cancer cases were diagnosed between 2017 and 2019. In 2020, the value rose to 8,950, which represents the highest case in a single year. To achieve early detection, automated systems have been proposed, particularly deep learning. It has shown to surpass the capabilities of radiologists, but they encounter a lot of challenges. For example, high computational costs, which affects, training and then using resource-constrained settings. Also, the challenge of data where to get training data becomes very difficult because it requires high expertise annotation, and also you need large volumes of data to train the algorithms. For our study, we aim to achieve a early detection whereby we can design a system that is less computational intensive, and then we can train that on a very small amount of data using, for example, a case hospital scenario, and then we can also achieve results that are similar to oncologists, to radiologists, or even better than results from radiologists. Basically, we are not designing to, take the jobs of the radiologists, but at least to, give them informations quickly about the analysis. To perform our experiments, we designed several, methods, which some of them are novel, and then we compare our methods with existing architectures. For example, AlexNet, GoogleNet, ResNet, MobileNet, and also some transformer architectures such as a Vision transformer and the Swing transformer. For the novel technique we developed, the Mavid pooling, we compare with the standard pooling techniques, which are the max pooling and the average pooling techniques. The experimental setup, we use a PyTorch running on a Jupyter notebook, and then we train the system using an NVIDIA GPU. We use the AWW optimizer. We use cross entropy loss function. We use the step LR scheduler, and most cases we use a batch size of 16. For our statistical analysis, we evaluated the model on accuracy, specificity, sensitivity, and area under the receiver, operating character receiver curve. We also performed PIT tests to test significant difference. We developed a novel metric, which is the cumulative scores given by that equation to tell us, for example, if we have 3 different models or 3 different techniques, which is the better technique, over several experiments. The first method that we proposed was the random pixel swap data augmentation, which we published in this journal. What does it do basically? Because we know that unlike conventional images where you can go to Google, for example, if you want images of cats, it's easy to get millions of images of cats, but when it comes to medical imaging, it becomes very difficult because of the high specificity that is required and accuracy to develop models like that. So because of this, data is very scarce for training lung cancer models. So we developed a method that can augment the data, which we call the random pixel swap data augmentation. What it does is that it's a patch-based technique whereby if you have an input image, for example, this image, we can then transform this image to provide meaningful representation of the data, and then we can achieve multiple images to train the model. Why we developed this was because available techniques, they fail particularly for, when you're using them for maybe CNN and transformer architectures, they do not generalize to both architecture paradigms. And also, unlike conventional training, if you're training a classical or conventional model, these techniques, they perform well, but when it comes to medical imaging, they fail, or in fact, they underperform and limit performance. So here is the algorithm that we developed for the random pixel swap data augmentation technique. Also, we compare it to state-of-the-art techniques such as cat mix, random erasing, mix-up, and cutout. These are standard techniques in literature. We evaluated the average training time, and then we saw that although our technique showed the largest training time, but this was not significant compared to other techniques. The difference was not significant. This makes our technique to be on par with the standard techniques in terms of training times. We also evaluated a technique for lung cancer detection, where we saw that our technique performed better than other techniques available in literature, which are the state-of-the-art. And using our cumulative rank score, we saw that our technique ranked the best, as you can see, rank 1 here for this particular task in detecting lung cancer. Also, we tested that on lung cancer classification. It also saw a similar performance where our technique, the random pixel swap, surpassed almost all the techniques considering the metrics. And this difference were actually significant. It is customary to train algorithm using segmented images because it has been shown that these images, if you segment the image, they can improve performance. So we tested to see if this technique used with segmented images can improve performance. And the result is presented here, where we see that our technique surpassed almost all the techniques, and it consistently ranked number 1 compared to the state-of-the-art techniques. Also, there are available traditional data augmentation techniques such as flipping and rotation. We also check that if we combine our technique with this traditional augmentation technique, does it improve the performance, or does it perform better than using this technique? In most of the cases, we see that our technique alone, surpasses the performance of the traditional techniques. And also, when we combine the traditional technique with our technique, in some cases it improves the technique, but in some cases our technique alone surpasses, combining these techniques with our technique. Reason because some of these traditional techniques, in fact, they reduce the performance of the model. So when you now incorporate our technique, it struggles, to improve performance due to the, loss in performance of the traditional techniques. Also, we validated our experiment because one of the problems of deep learning is if you train a model on a data that it is used to, for example, let's say George Macarius between Safaku Makato, you use the data there, you train the model on it, and then you go to Steve Biko Academic Hospital, and then you try to, implement the model to analyze, same CT scan data. You see that it fails drastically and woefully. So we try to see that can our technique actually, improve generation capabilities. So we take data from different, regions and different city protocols. We trained on one, and then we tested on the other. We saw that our technique, in fact, performed excellently compared to other techniques, and in fact, it ranked number 1 for all metrics with significant difference. The second technique that we propose is the Maverick pooling technique. What does this technique do? Remember, we want to develop an algorithm that is computationally effective, and then we can train it on relatively small amount of data, for example, that can be used in the clinical local hospital settings. In CNN architecture particularly, it has different layers, so we tackle the pooling layer. So what pooling does is that it selects, it downsamples the image through different mechanisms. The major ones that are available in literature, which are very common, that are used in architecture, such as the GoogleNet and the ResNet, is the max pooling and the average pooling techniques. But we see that when we apply this to city images, when the model gets deeper, it loses signals. Sometimes the model overfits the data. So we come up with this technique to replace the pooling layer in the CNN and then with the Maverick pooling technique. Basically, this is what the Maverick pooling does. If you have an input feature, for example, in here, you can see if you apply a 2x2 max pool, you get this value. If you apply a 2x2 average pooling, you get this. But if you apply our technique, which is the 2x2 average pool, you get this. And then we can see that as this goes deeper within the network, this signal might be lost so soon. And in fact, if you look at mass pooling, although it seems better than average pool, but compared to our technique, you see that this can still go further. For example, if you differentiate this, you still get a better feature that goes deeper down within the layer of the network. We tested this technique on city images, and then we saw that our average pooling performed better than max pooling and average pool. We also developed a custom, cost-effective CNN network where we replaced, you can see here, you have the convolution layer, the batch normalization layer, and then here, traditionally, is always the pooling layer. We replace this pooling, which is usually max pooling or average pool, with Maverick pool. And then the other layers takes the conventional CNN architecture. We saw that our Maverick pooling technique, when we introduce it here, provide better performance. Also, to validate the approach, we consider standard or state-of-the-art models that are available. We use the AlexNet, ResNet, GoogleNet, and then we change or replaced the pooling layers of this network with Maverick pool. We see that when we replace that, the model performs superiorly better. For example, if we consider GoogleNet that uses the max pooling layer, and we replace it with Maverick pool, we get an improved performance here. In fact, a very interesting case is the AlexNet. You can see that the difference, is very significant and is high. And also the same with ResNet. It shows that this Maverick pooling provide a better feature extraction, mechanism compared to max pooling and average pooling. Also, the third thing that we did was published in this journal. Basically, what this study does was to propose a model that used an ensemble technique. And what are we trying to achieve here? For example, in the hospital, lung cancer is basically, diagnosed and treated by, a lot of specialties. You have the radiologists, oncologists, you have the medical physicists, and, a lot of them that come together, for the patient treatment. So we simulate that scenario that if we can have different models coming together to, analyze the data and come up with, a final diagnosis, it might present a better solution than using a single model. And then we tested that approach. We developed, this algorithm flowchart where we have, we use the input. Here is the input image, the city image. We perform image segmentation of the region of interest, and then we pass this into the 3 different models where you see this is the model 1, model 2, model 3. We are trying to simulate, for example, 3 doctors who are analyzing this scan, and then they come up with different, diagnoses. We evaluate each of the diagnoses, and then we assemble them to form a single diagnosis. We evaluated that, for example, in lung cancer detection, we saw that if we look here, the proposed model that combines the 3 models is the deep nodal detect that we proposed, and then these are the results of each of the models. We saw that the ensemble model at every case provided improved performance here, and here. Also, same as when we try to classify the lung cancer, after detection, then we can say, oh, there's lung cancer here, but what type of cancer is it? For example, adenocarcinoma squamous cell, we try to, make the model to classify the data for faster, diagnosis. And then we see that the assembled model at every instant surpass, the individual models here. Another interesting thing that we observed was that if we augment the data, the performance improved. If we apply segmentation of region of interest, it improved performance also. And here we can see the table of confusion here that shows the confusion matrix for each classification. We compared our study or result with previous studies that have analyzed the same data set that we analyzed. For example, here we saw this study, Karim et al., Suleiman et al., and we see that ours using this data set provides or gives the best results. And also for this data set, the chest CT scan data set that we employed to classify lung cancer, to date in literature, our results remains the best, achieved accuracy in this regard, as of today in literature. In conclusion, our study demonstrates that, in fact, we can use a low-cost deep learning model to effectively, diagnose lung cancer using patient CT scan. We also saw that if we utilize ensemble technique, data augmentation, segmentation of region of interest, the performance improved. In the future, we aim to extend this study to other imaging techniques such as the MRI, ultrasound, and X-ray imaging because, for example, MRI and ultrasound, they provide or introduce less ionizing radiation risk to patients compared to CT scan. And what does this imply? The implication means that now, with lower computational cost, local hospitals can develop deep learning models or automated detection and diagnosis system in their hospital, not relying on high performance computing, which a lot of hospitals cannot afford. And also we saw that our method was able to solve the problem of requiring large amount of data to achieve performance. So with limited data in your hospital, we can design a very cost-effective model for you that can run on, the normal computer or normal machines, and then we can achieve significant and improved diagnosis. Our code is available in GitHub. You can check it, and it's freely available as long as you cite, the publications. We appreciate the CSIR for their financial support. Here are the references that was used for the presentation, and thank you.
Thank you very much. A very needed study, very interesting. Do we have any questions? Yes, please go ahead.
Hi, thank you for your very interesting study.
Thank you.
I just wanted to ask, how early can your model detect lung cancer? How early on is your model able to detect lung cancer?
You're saying how early?
Early, yeah.
Yeah. For us, study shows that as early as possible, let me use that, because we do not know when the patient presents at the hospital. We based it on the data that we analyzed. So if you have 100 patients presenting, they must have presented at different timelines of the cancer stage. And then for our model to be able to, achieve almost about 99 to 98% detection accuracy, it shows that it can detect the cancer at the earliest stage. And then we saw through our analysis that there are, patterns in the image that are not visible to oncologists or radiologists. We tested that. You give an image to the radiologist, and then they tell you this is a normal scan. And then we put it to our algorithm. It tells us this scan is cancerous. And then we compare with result of biopsy results, and then we see that it is true. So it means that our model can detect lung cancer earlier than radiologists can. So this actually is what makes the study very interesting, that it can be, practical for hospitals to, have models like this. Although, like I said, if you look at this slide here, it is not replacing the radiologist. You can see the AI here, it's informing the radiologist. You know, there's a CT scan, it's been processed by our algorithm. The AI tells the radiologist what it sees. Then, you know, within a very quick time, the radiologist can confirm, you know, what the AI have seen. And it makes the job very fast, effective, and then we can achieve more accurate diagnosis, because in the hospital, a lot of time, false positive are detected, and in fact, even false negative, where there's a patient that comes in, for example, in routine. You know what they do now, for example, in advanced country, they do routine lung cancer screening, where people who are at the risk of lung cancer, for example, who are smoking, they come for that screening, and then they use automated diagnosis system to analyze the data, and they get, you know, good result. A lot of people were diagnosed from, you know, studies like that, for example, in the UK, and it has helped to improve survival, particularly for lung cancer.
Thank you very much. I can take one last question. Yes, please, Prof.
Thanks, Ibi, for that presentation. Just the first one, how do you ensure that you're not overfitting that model to reach the ranks of 99% or whatever it is?
Okay, for us to be sure that we didn't overfit the model, we employed several metrics, like I said, sensitivity, accuracy, and AUROC. But the killer method that we used to know was the one that I described in terms of generalization capability, where we trained the model on a data that was acquired with an imaging system, and then we evaluated with data that were acquired with a different imaging system of different population, in fact, in 2 different continents. And then we saw that, in fact, the performance was consistent compared to, you know, other methods. So that tells us that our model is not actually overfitting the data. It is learning the true representation of what it is to be lung cancer.
Last one. So in all of these models, you use what, 80-20
Pardon?
ratio? In your models, what ratios of trained tests?
Training, we use 70-30.
70?
Yes.
30.
30 for training and then 30% for test,
okay,
which is the standard in deep learning mostly. Basically.
Okay, thank you very much.
Thank you so much.
Just give him a round of applause.
Okay, our next presentation is by Toboane Ramolongo, who is exploring the synergy between traditional medicinal knowledge and modern computational science. So her official title is Integrating Indigenous Medicinal Knowledge with Computational Modeling for the Discovery of Antiviral Drugs. Thank you very much.
Good morning. I just need to make sure that I don't forget to breathe. Just looking at the title, you can see that this is a very ambitious project. It is a multi-disciplinary project that touches on indigenous medicinal knowledge. On that alone, we need an understanding of the indigenous uses of the plants, which then it brings in the social, cultural aspects, and then the use of medicinal indigenous plants. Then it brings in bioprospecting and metabolomics. The second thing is the computational modeling for drug discovery. Then it brings in molecular and structural biology, artificial intelligence, and machine learning. And another thing is virology. So you need to be able to understand the intricacies that are associated with the viral life cycle. My name is Toboane Ramolongo. I am a researcher with a PhD in microbiology. with expertise in virology. So I am based within the Indigenous Knowledge System program under the Advanced Food and Agriculture cluster within the CSIR. So I will just skip through this, then you'll catch up as I go through the presentation. Just to give a background on IKS, Indigenous Knowledge System is a very broad term, so it touches on the knowledge, the skills, the know-hows, the technologies, the innovations, amongst the others. And this is the knowledge that have developed within the communities for centuries, and it has been passed down from generations to generations. So the communities, they have used this knowledge to adapt with, so that they can strive within the natural environment. And then this IKS is cross-cutting, and then it finds applications in agriculture, in health, food product preparations, and environmental conservation. So with environmental conservation, it was interesting to find out how they do this. So they use taboos, like let's say for plants, if they see that the plants is starting to be becoming extinct, then they start creating stories against that so that people, they don't use it that much. So I am within the IKS program, so we are mostly focused on medicinal plant species, so we do mostly the validation of the indigenous knowledge so that we can substantiate their claims and also develop products. So the importance of IKS is seen on how from how the public is interested in this field, and that is reflected by the legislations that have been placed to protect the knowledge and also the indigenous genetic resources. So one of the acts or the legislations that is in place in South Africa is the National Environmental Management, specifically the Biodiversity Act. So this act is enacted to protect the indigenous knowledge, and also it's enacted to protect living organisms. It can be animals, it can be plants, it can be microorganisms. So if we are doing any activities that's associated with naturally living organisms, then you activate the compliance of the act. So with the IKS, there's this one regulation that emanates from Chapter 6 of the Biodiversity Act. So it is the bioprospecting, access, and benefit sharing. So it says that whenever you do research that associated with the natural resources or with indigenous knowledge, you need to make sure that you are compliant. So when you do research, you go collect a plant, you need to make sure that you notify the Department of Forestry, Fisheries, and the Environment that you're doing this and this with these resources. And then once you come to a space where you want to now patent or you want to commercialize your product, then you activate the commercialization phase permit system. So this part of the commercialization phase has taken us, the IKS team, to the bundles where you find yourself with a suitcase filled with long skirts, because now you need to adjust to the cultural expectation of that area. And this is the last act, so is the Indigenous Knowledge System Act. It has a very long name, which was signed by the President in 2019. So this is a sui generis act which strictly protects IKS. You can see that IKS space is highly regulated. So likely for us at the moment, although this act has been signed to law, it's not yet enforced. So I've mentioned that the indigenous knowledge is passed down from generation to generation. Mostly you find that this knowledge is living knowledge, which means it's orally transferred from the generation to the generation. So what the GSTI, and in collaboration with the CSIR, what they did many years back, they created this system, the NICMAS,
which is, so in this system, it is a recorder system where you find all the information that you need on plant species, and then you find also the communities that are associated with the traditional uses of those plants. So it actually is very useful for us researchers, because then you can go to communities to source this information. So you can actually find all the information regarding the traditional uses of certain plants. But at the moment, it's very restricted, so you can only access certain information, but you cannot access the details of the communities. So it is still an ongoing project where they're still documenting this information across the country. So from this system, we were able to identify 62 plant species that were the communities are using to treat respiratory infections. And the another thing when it comes to IKS, let's say with, okay, I'm going to use COVID because then it's more related with RNA viruses. When you have a COVID-19 infection, then you, as the communities, they don't say that we are treating this disease. They are more treating symptoms. So when you work, even from the recorder system, you find information based on symptoms than on the actual disease. And then I mentioned that this project has also the antiviral aspect to it. Then the question is why there's so many diseases, why then you choose viral infections. So WHO, the World Health Organization, time to time, they update that they publicize the list of priority diseases that the research community needs to focus on to develop diagnostics, therapeutics, and vaccines. So this list of pathogens or diseases, they are selected based on their possibilities, the potential to cause the next pandemic. And after the COVID-19 pandemic, then the WHO decided to also add the disease X, which was never in the list. And then the disease X is there to denote that the next pandemic might be caused by a pathogen that we have not encountered before. So if you take a closer look to all these viruses, you find that they are caused by, but these diseases, they are caused by RNA viruses. And then most of them are zoonotic, which means they are transferred from animals to humans. Just to give more of a picture of how often we encounter the spillovers or the emergence of these viruses over time, you can see this is the timeline infograph that shows the spillovers of these viruses from, this is over two decades. You can see that most of them that are here, they are listed under the WHO list. So there's Ebola, CCHV, and the Lassa virus, Zika. So most of them, they are listed as the viruses that still needs the research communities to develop diagnostics or therapeutics. So the one thing also that I want to mention with these viruses, the one thing that's playing a role with them being emerging or re-emerging is the global warming, and also the animals they're finding themselves being displaced from their natural habitats, and the people are also interacting more with wild animals. Then this assists to the spillovers, the transfer of these viruses to humans. So we really need to prepare ourselves for anything that's still yet to come. So this is more of a strategy that we want to use towards in this project.
when it comes to drug discovery, there's a conventional way and also the in silico modeling way. So I'm a person who don't like things that takes time. So with a conventional way, you need to have a whole lot of plants that you need to screen, and then you can see down here. So you need to first identify the target, and then you do all the screening and everything in the lab, which is slow. But then with the in silico modeling, it's faster. So I've mentioned that we are using indigenous knowledge. So what's happening with indigenous knowledge, it guides us to select plants, because you don't want to just go out there and take everything and then bring in and work with it. So indigenous knowledge guides us to streamline the plants that we can work with. And then we use the chemical profiling using the LC or MS, LCMS or GCMS to create the profile from the plants. And then so with computational modeling, then you will need the compounds from the plants, and then you also need a target protein. So I'm going to explain a bit more on the target protein, why we chose the specific one that we are working with on this project. So you can see with the in silico modeling, once you have the ligands and then the target, then you can run all this in the model, using different models, of course, and then you can have heat compounds that then you will bring in the lab, and then do your cytotoxicity and all that before then you go to in vivo. So it kind of like streamline the process that you need to actually get a therapeutic product. Okay, so I've mentioned that the diseases that are listed by WHO, they are caused by RNA virus. Most of them are caused by RNA viruses. So I remember during the COVID pandemic, one morning we just wake up and then we'll be told, oh, there's another variant that's spreading like fire. And this is because that the RNA viruses, as they make copies, as they replicate, they create mistakes which makes them, which then create new variants. But then also the virus has some proteins that it actually needs to survive. So it has to make sure that those proteins, they do not change for it to replicate. So the RNA viruses has this protein, which is the RNA-dependent RNA polymerase. So it helps the virus to replicate. So this is the structure of the RNA-dependent RNAs, and then it has an active site. So that means then when you look for therapeutics, if you can block this site, then the function of this protein won't be effective, and then the virus will not replicate. So, and then I've mentioned that this protein is present in all RNA viruses. So once you have a drug that works against this protein, then you can prevent the infection of all RNA viruses. So that's why we chose this one. So for this study, we plan to follow a very straightforward approach. So this part of the project is something that's still very new to me. I'm a virologist, but I'm excited to learn on this part of the project. So we planned to create a database of compounds that are present in the plants that we have chosen, and then from there we will do a molecular docking analysis with the RNase-dependent RNA polymerase as our target protein and the photocompounds as ligands. And then from there, we follow by doing ADMET analysis. ADMET analysis is just to check the druggableness of the compounds, and then followed by the molecular dynamic simulation, which then determines the stability of the complex, because then the protein and the ligand and the target needs to make a complex so that the protein does not function anymore. So you need to check also the strength of that interaction. So after you do all these things in the computer, you can say that, okay, I have my drug, because then you need to still go back to the in vitro, do your in vitro studies, and then check if this is the true picture that you have within the biological system. So this pretty much summarizes what we need to do. So I have one case study, like one. We did more of a prelim evaluation of this. So we chose this plant, Kigelia africana, is also an indigenous plant. So for this one, the compounds, they're already well documented, because that's another problem with indigenous plants. The knowledge, the information, the data is very limited. But for this one, we're able to get about 152 compounds from the literature. So we did the molecular docking simulation. So another thing with the RNA-dependent RNA polymerase, the source structure of the protein is readily available, and then what you need to do is just to prepare the structure of the protein, and then you clean up your ligands, and then you do the docking. So this is one of the results that we did once we ran the molecular docking. So we were able to find two compounds that showed high docking scores, and the docking scores, they were compared to remdesivir. Remdesivir is a FDA-approved clinical drug that is used against the RNA viruses. So you can see that the compound 1 shows like -11, and then compound 2 a bit lower, but -10, which is almost in the range of remdesivir. So this is the prelim work. So we still need to do the ADMET analysis, molecular dynamic simulation, and then the in vitro studies to confirm if this is the true picture that we get in vitro. So basically, this project, where we want to take this project, we want to prepare ourselves for the next pandemic. So we have rich knowledge of indigenous. We have rich indigenous knowledge and also the diversity of medicinal plants. So we want to create a database where we can actually have it readily available, and also feed back to the NICMAS, the recorder system, so that at least once it gets open to the public, then you can see also the scientific information that's associated with those plants. And then also we want to be able to have a whole process from the database, and also from the modeling and the in vitro, that we can actually just pull out if then in case of the next pandemic, because then if you have these plants growing, then the communities can just use that to treat whatever virus that you have. And then so with IKS, whatever we do is linked to communities. So that means at the end of the day, if we discover a drug that can be commercial, so that means then the communities that are associated with that, they will also benefit. So that will be our end impact. So we have a product that also impact the community. Thank you.
Thank you very much. Very interesting. I actually appreciated you spending time on the X, because there is that sensitive line that we need to toe between research and indigenous knowledge. I'll be taking a few questions. Oops, sorry. That's my time. It doesn't want to stop. Any questions? Please go ahead.
Thank you, Dawani. That was very interesting. I'm your colleague, but every time I hear that, it's very interesting to see how IKS can be even integrated with like computer systems. So on the microbiology side, you mentioned something about docking. Is that when you bind like the interaction that you have to limit the growth of the protein?
Yes. So like with the plan is that the way some therapeutic works, so you have a virus. So virus produces a whole lot of proteins when it replicates. So you just choose which one you want to focus on, which is then to the target protein. And then you have your compounds. So you want a compound that will be able to bind to that protein, so that that protein doesn't do its function anymore. So if it can't function, that means the virus will not be able to replicate anymore. So that's the interaction that we need to establish.
Okay. So how how does the binding happen? Like, is there a certain criteria for the compounds to actually bind with the protein? Like, how do you know that? Or is it a trial and error kind of, or you have to?
So for this one, I'm not sure if I'm going to answer that well, but we are using Schrodinger Maestro. So that's the program that is developed for drug discovery. So it does have all, so you can actually set it to the way that you want. Like you can set the limit if then you don't want the interaction. If you want it to be in this range, then you can set it up. So that's within the model, which I am not very clear.
Oh, okay.
Yeah.
No, thank you.
I'll just take two more questions. So I'll start with the lady there, and then Paul after. Then.
Thank you for such a great talk. I wanted to find out, since you mentioned that you targeted the active site, did you also maybe consider blind docking, especially if there is allosteric inhibition?
You see, can I have a conversation with you after this? Yes, that's all I can say. Yes, I still need a bit of more help when it comes to the modeling. Yes. Hi.
My question will practically be a tad bit off course.
Sorry, say that again?
My question will be a tad bit off course, but it's
Okay.
related to what it is that you're doing.
Okay.
You are specializing on IKS. So when you were gathering the information, are you necessarily only basing this on anecdotal evidence? Do you include, or rather, did you, in the process of gathering information, consult, say, maybe the traditional healers organizations? Because if I'm not mistaken, some of the cures that you guys are actually either discovering or attempting to prove their validity has already been discovered by the traditional healers, more so because it's indigenous knowledge.
Yeah.
Did you by any chance consider or contemplate roping them in? Secondly, would it also be possible for you guys to integrate what they already have into what you are developing to actually facilitate for either the comparison thereof of the results that they get that have been tried and tested by time, vice versa, the modern day sciences and methods of medication that are being conducted and used today? Thank you.
Yeah, that's a very...
10 seconds to answer.
No, it's actually not. Because when we do community engagements, it's mostly when we are at the commercial phase. So that means at that point we've already done the research and everything. And most of the concern that they raise is that they would have loved for us to consult them at the beginning so that they can be part of the process. But the challenging thing with the AKS is that most of this knowledge is oral. And then at the beginning of research, you just maybe find from a publication that there's this use, the traditional use of these plants for this and that, and then you do the scientific validation. So you will only be required to have the beneficiary agreement when you do the commercialization phase. That means then you have to go and then consult those people to have the agreement. But at the beginning of the project, like I said, the plant species that I have identified for this project, I found them from the NICMAS, from the recordal system. And then already on that system, you can only see limited information. Like the communities, the JSTI is still withholding that, so you cannot get. So you can only get that these plants that is traditionally used for this and that. And then we will do the research. And then if then we find information that is like we confirm that is true, and then you can actually have a product. That's when you will be required to have that agreement with the knowledge holders. But at the beginning, it's very difficult. But it's something they will prefer for us to do. Yeah.
Thank you very much. Paul, can I just ask you to take your question offline? Apologies for that, but we are running over. Oh, Pinda, are you feeling sorry for him? Yes. So in the morning, so can you please give her...
In the morning, we were told that this is a nice and friendly environment, so please forgive us a little bit for running over time. So our next speaker is Matapelo Motsau, whose research is focused on the nuclear latifolia, and the official title of the presentation is Evaluating the Effect of Nuclear latifolia plant extract against the Candida albicans virulence. Virulence, yeah. Good day, ladies and gentlemen. My name is Matapelo Motsau. I'm a master's student from the University of the Witwatersrand, and I will be presenting my research titled Evaluating the Effect of Nuclear latifolia against Candida virulence. Okay, so Candida albicans is a commensal microorganism that is found in the skin, in the oral cavity, in the gastrointestinal tract, as well as the vaginal tract. It exists in different morphological sites. So there's a transition in morphology of Candida. As we can see with our picture at the top, we have the yeast form, the pseudohyphae, and the hyphae form. So the pathogenicity of Candida albicans is also influenced by a number of virulence factors, such as adherence, biofilm formation, the formation of germ tube, and also the production of hydrolytic enzyme. And due to this, Candida albicans is able to cause an infection that is known as oral candidiasis in the oral cavity. Sorry about that. Okay. So with my research, because of Candida that can be found at various sites in healthy individuals, my research focused mostly on the oral cavity. So what happens when a person is infected with Candida albicans? The microorganism attaches to the epithelial cells of the host. Once that happens, this process is known as adherence. Once this happens, biofilm is able to form, and in the same length also, we see the formation of hyphae. So the importance of hyphae in the infection is that the hyphae is able to allow the microorganism to actually penetrate into the epithelial cell of the host. Once the organism has penetrated in the mucosal membrane of the host, we now see the production of hydrolytic enzyme. And these hydrolytic enzymes are namely phospholipase and proteinase. So the importance of these hydrolytic enzyme is that they degrade the phospholipids and the proteins that are essential for the survival of the human cell of the host. So once this has happened, once the infection, the microorganism has invaded the epithelial cells, it is now an infectious disease. Now the infection is progressing, and we see in most people who are infected with oral candidiasis is that we see oral thrush in their mouth. So the common name for oral candidiasis is oral thrush. This is because of the overgrowth of candida in the mouth. And usually this happens in people who are immunocompromised, people who have weakened immune system. And we also see it mostly in children. because we know that children obviously have weaker immune system compared to adults. So currently, there's quite a limited number of antifungals that are present for just overall the treatment of fungi. And these antifungals are mainly the azole and the prolines, right? But the challenge with the present antifungals that are present is the emergence of drug resistance. So yes, we have drugs that are available, but they are limited. And also, something that I can also add is that when compared to bacteria and viruses, fungi has quite a limited number of antifungals that are already available in the market when compared to other microorganisms. So now, in addition to that, we add that there's an emergence of drug resistance. Hence, there's a need for the development of new therapeutics for oral candidiasis. This then leads our research to actually look into medicinal plants. The reason why we are looking at medicinal plants is actually because of they have been used previously traditionally by Asian folks, and also it's said that they do have bioactive compounds that are able to treat different kinds of diseases. Hence, we are looking at medicinal plants. One of the plants that we actually looked at in the studies is Nuclear latifolia plant extract, which has been used traditionally to treat dental cavities and oral sepsis. And also research has shown the potential of this plant used at the different parts of the plants have been used to test its potential in antibacterial, as an antifungal, and also as an viral. So what has not been done actually with this plant is to test its potential as an antivirulence. So the reason why we are looking at the antivirulence is because of, as I mentioned at the beginning, is that Candida albicans is a commensal microorganism. So we still need it in the body, though it is an opportunistic pathogen, but we don't want to kill it. We want to inhibit its virulence. So hence the need to actually be able to inhibit the virulence that occur when the infection does occur. So this leads me to the aim of my study, which was to determine the antimicrobial effect and the antivirulence potential of Nuclear latifolia against Candida albicans. The objectives were to determine the antifungal effect as well as the antivirulence effect of the mentioned virulences, and also investigate the effect of the plant extract on the virulence genes of this microorganism. 5 clinical strains of Candida albicans and an ATC strain was used, and all the experiments were repeated in triplicates. Ethical clearance was obtained for the use of the fungi of the Candida albicans, and as well as the use of the epithelial cells that were donated. For the methods, the micro broth dilution was used to determine the MIC, which is the inhibitory effect, and also the fungicidal effect of the organism. Once we got the MIC, then the MIC that was obtained was further used to determine actually the effect of the plant extract on the adherence of the fungi to the epithelial cell, the formation of biofilm using the crystal violet method, and germ tube formation, the production of proteinase using BSA agar because it's able to produce nitrogen, phospholipase production using the egg yolk, and also the gene expression was determined using qPCR. For my results, the MIC that was obtained was 0.16 milligrams per mil, and the MFC was 0.63 milligrams per mil. The MIC that was then obtained, which is the inhibitory effect of the plant extract, was further then used, as mentioned, to test the virulence factors. So it was used at the concentrations that are here. For the adherence assay, the percentage reduction that was seen was up to 85%, and at the lowest concentration which was used, which was 0.04 milligrams per mil, it was 73%. And the significance of the plant extract being able to inhibit adherence is that adherence is actually the first step of the infection. So if ever the plant extract is able to actually inhibit the first step of the infection, it means that there won't be any progression of the disease. For the formation of biofilm, this was done at 6 hours. So according to literature, biofilm of Candida albicans is able to form by 6 hours. We see that the biofilm has formed. So we saw a percentage reduction of up to 50%, and at 6 hours, and also at 24 hours, we saw a percentage reduction of up to 40%. And these were at the higher concentration of the MIC that was 0.16 milligrams per mil. So with biofilm, it is the reason also behind drug resistance is because of biofilm. So we can see from the results that it becomes very tricky to actually deal with the progression of the infection once the biofilm has formed, because of biofilm actually have extracellular matrix, and these extracellular matrix protects them from the antimicrobials that might be given to the host. So it is quite difficult to deal with biofilms in various microorganisms. It's not only in Candida. Another thing that was observed is the percentage reduction of germ tube. So as I've said previously that Candida is able to exist in different morphologies. So the germ tube, which is the hyphae, it's actually the most pathogenic form of Candida, and it therefore assisted in the penetration to the epithelial cells of the host. So we saw a percentage reduction of up to 51%, and so if ever the reduction of the hyphae is, if ever like the hyphae is reduced, it means that there will be less available hyphae that are in the host that will be able to penetrate into the cells. So hence it was important for us to see a reduction in germ tube. Okay, and then with proteinase, there was no effect that was seen or observed when we tested the proteinase. And the proteinase are actually enzymes that degrade the proteins in the cell membrane. So this shows that once the microorganism has invaded in the host, it becomes quite challenging to deal with it, because of now the infection has already progressed. So that we could see that there was the plant extract was not able to prevent the degradation of the host cell, because of it had already invaded the host. For phospholipase, we also saw that there was a low and moderate activity of the low and moderate activity that was observed of the reduction of this plant. And the PZ value are actually we are measuring the precipitation zone of the effect of the thing of Candida albicans. So we could see that at 0.76, that's actually a low enzyme activity that was observed. So when we can see that it is able to block the secretion of phospholipase. So phospholipase, when compared to proteinase, it's able to be blocked. Because of proteinase, we could see that it was not able to be blocked by the plant extract. On the gene expression assay, we saw a downregulation on ALS3, which is a gene that is responsible for adhesion, and HWP1, which is a gene that is responsible for hyphae formation, PLB1, which is responsible for phospholipase, and the sub-genes that are the proteinase. So the reason also behind us doing the gene expression, it was to validate the results that we obtained in the other assays, that is it really, when we check the genes, the virulent genes, is it really having that same effect? And we could see that it was able to downregulate all the genes that are expressed. And one thing that we could see was that on proteinase, one of the proteinase genes, which is SEB3, there was quite an upregulation when compared to the other SEB genes. It's because of there are a number of SEB genes that do take that play a role in this microorganism. So the upregulation might be that there's literature that still states that sometimes when you introduce antimicrobacterial when you introduce an antifungal into Candida, what it does is that it actually initiates, the microorganism becomes more excited. So instead of it actually being suppressed, it actually goes up. Hence, we can see the upregulation in SEB3. To conclude, from my results, we can see that N. latifolia is able to inhibit Candida albicans. It has an antifungal activity. And we also saw that its potential in antivirulence effect in adherence, phospholipase, hyphae, and biofilm. And we could see this, we can confirm this by the downregulation of the virulent genes. So what can be done is that we can now start doing more investigations in terms of the cytotoxic effect of nuclear latifolia, because of what we would not want is this plant extract to be actually toxic, because of we know that fungi is actually a eukaryotic, and also mammals, humans actually are also eukaryotic. So what could be done further is to check its cytotoxic effect on human cell lines, and also to characterize the bioactive compounds that may be found in the plant, because of I did not go through the process of characterizing the bioactive compounds that are actually in the plant. And once this is done, then the plant extract can be taken into further and being tested in animal models to see how effective it can be, so that it can be able to help individuals that do have oral candidiasis, because of most of the people who have oral candidiasis and are immunocompromised find it difficult to eat and to chew. And if this is the case, they won't be able to take their medication for whatever that they might be struggling with outside of oral candidiasis. These are my references, and I would like to thank my supervisors as well as the CSIR for giving me the opportunity. Thank you very much.
Thank you. So I'll be taking 2 questions. Do we have any questions? Yes, please.
Thanks for that presentation. May you please go to slide 2? I mean, not slide 2. Yeah, figure 2.
I don't know what I did there. Can you maybe start with the question while she finds the slide?
I want to reference the figure. There, in any case, we have those concentrations at the bottom that control there's concentrations up to 0.6. But you can't get to the figure.
Sorry?
You can't reach the figure.
Which figure? Sorry.
Figure 2. Not slide 2, figure 2. Yeah, there. No, yeah, even there. So in that trend, 0.16, what would have happened if you had gone further? Would that decrease?
If what? I don't understand the question.
On your y-axis,
Okay.
the concentration is at 0.06, what? I can't quite read that thing. But on the x-axis, you've got a concentration that is increasing.
Yes.
The bar graphs are also decreasing. So I'm asking, if you had gone further,
Okay, yes.
would those bar graphs continue that trend? In other words, there is a negative linear, actually.
I really I don't know whether I'm not getting the question, but you're saying if ever I was going downwards, meaning
If you had continued increasing that concentration.
Yes, oh, okay, now I get it. So the more we increase the concentration, the more effective it would be, if I'm getting what
So
you're
is
saying.
there data demonstrating what you're saying? Have you gone further than what you've reported?
No.
One more question.
Thank you, ma'am. Nice work that you're doing. It's very relevant. So this is just a question of more of, if I was an entrepreneur, how would your work give me like a market advantage?
How would my work give you a market advantage? Okay, so because of oral candidiasis affects people that are mostly immunocompromised, right? So let's say that we have a lot of people that are sick because of once the infection has progressed, there are high chances of the patient actually dying, right? So if ever we are able to prevent these people from actually getting to that stage, and these people can be people that are employed in different industries. So imagine now you're having a lot of your people, because of whatever diseases that they might be facing, and then oral candidiasis occurs as an opportunistic pathogen. So it means that now you will be losing, a person, an employer might be losing people because of people are actually sick in the hospital because of there's no treatment for this infection and stuff like that. So as a marketer, you know that people will need to buy this medication so that they prevent the progression of the disease. I don't know if.
No, very much. Thank you. Thank you.
Thank you very much. Can we just give a round of applause?
Okay, so we are almost at the end. Our final speaker is Hale Letso Otibeng, whose work highlights the pharmacological richness of local flora and also demonstrates the power of in silico profiling in modern natural product research. So the title for her talk is Computational Metabolomic Profiling of Anti-Venom and Anti-Inflammatory South African Medical Plants. Please come through.
Okay. Let me tell you in just kind of how it works down arrow for next, up, previous. This is your printer. Okay, got it. Thanks. Okay, good day, ladies and gentlemen. As introduced, my name is Khaleledzo Utibeng. I'm a PhD candidate at the University of Johannesburg under the supervision of Dr. Fidel Tugzimana, Dr. Zamani Cele, and Prof. Justin van der Hooft. And today I'll be briefly sharing on my ongoing project, which focuses on the use of computational metabolomics to profile or elucidate anti-inflammatory and anti-venom South African medicinal plants. Now, medicinal plants have been used for centuries for the treatment and management of a wide array of disease conditions as well as ailments, ranging from colds and flus to skin diseases, malaria, cancer, amongst many others. And South Africa is actually one of the countries with a long history of the use of medicinal plants, as well as a rich biodiversity of these plants, with over or about 60% of the South African population relying on medicinal plants as their primary health care option. Now, the therapeutic and health benefits of medicinal plants can be attributed to the bioactive compounds that they possess. Now, these are also termed plant-derived natural products, and they are continuously being explored for the development of new drugs. Now, there has been indeed multiple drugs that have been developed from plant-derived natural products. A typical example is artemisinin, which is isolated from the plant Artemisia annua and is popularly known for its use as an anti-malarial drug. Now, previously, there had been a significant decline in interest in natural product research, particularly from medicinal plants, and this was owing to several reasons, such as the perceived long discovery and development process, complexity in the synthesis process, as well as the high frequency of re-isolation of known compounds and difficulties in elucidating the chemistries of these traditionally used plants. However, the past few years have seen increasing or a resurgence of drug development from plant-derived natural products, and this is driven by advances in the technologies as well as the development of strategies that help us accelerate this discovery of plant-derived natural products for drug discovery purposes. Now, South Africa is well positioned on a global landscape owing to its rich biodiversity of these traditionally used medicinal plants. Two examples of these are Melianthus comossus and Hypoxis hemerocalidea, which are the plants that are under study. And these, although they have many uses, we are mostly interested in the uses for the treatment and management of skin diseases and conditions, as well as snake bites. Now, Hypoxis hemerocalidea has also been known, or is popularly known, as African potato, and this is due to the fact that the shape of its bulb mimics that of a potato. Now, the two plants were selected, number one, based on claims of the anti-inflammatory and anti-venom activities or benefits that they possess, as well as based on their availability and accessibility. Now, with regards to how exactly these plants are used on traditional medicine practices, the roots and the leaves of the plants are normally boiled, and then the decoctions or the pastes are then applied on wounds, swellings, and burns in order to promote their healing. Baths with the herbs have also been said to relieve joint pains, while the roots of Melianthus comossus have been used in the treatment and management of snake bites. Now, despite the many uses of these plants, there is, however, a limitation with regards to knowledge on the phytochemistry of the plants. Now, this speaks to what is in these plants, as well as the underlying biochemistry, which speaks to the specialized metabolisms of the plants, and more simply put, which compounds in the plants are responsible for the benefits that we see and experience. Now, omics sciences such as metabolomics offers unique opportunities for us to be able to comprehensively profile the chemistries of these plants and better understand the underlying biochemistry of these plants. And this is an advantage towards natural product research for drug discovery and development purposes. Okay, so what is metabolomics? So it is a system biology discipline that involves a qualitative and a quantitative analysis of the metabolome. Now, the metabolome is basically all the metabolites that are in a biological system of interest. That can be a cell, a tissue, or an organism as a whole. Now, this is just to show some of the applications of metabolomics in natural product research, as well as some of the work that has already been done in this regard. Okay. Now, despite the maturation of the field of metabolomics over the past decades, and despite some of the technological advancements that have been made regarding the analytical platforms that are used, identifying or annotating these metabolites still remains a great bottleneck. Now, this then limits the biological insights or information that we can generate from such studies. Typically, only about 1.8% of spectra that is generated from metabolomic studies can be annotated, meaning we can put names to these detected chemical signals, while the rest or majority of them still remain unknown, falling under what is called the chemical dark matter. Now, computational tools or computational metabolomics has then been proposed as a solution to help us overcome these limitations of metabolite annotation and identification. Now, given the artificial era that we are currently in, the past few years have seen efforts from the scientific community to develop as well as apply several computational tools and strategies that will help us decode these chemistries of medicinal plants even further, be it at a structural, substructural, or even at class level, helping us know more than we would using the traditional approaches. Now, this is just to show some of the computational tools that we are exploring and applying in this particular study. Okay, so the study had three main aims. However, for the purpose of the presentation, I will be focusing on the first one, which looked at elucidating the metabolic landscapes of the two plants under study, Melianthus commosus and Hypoxis hemerocalidea. Experimentally, the following steps were followed. So the plants were purchased from a nursery in Mildersdrift, and harvesting of the leaves and the roots of the plants were done. So we focused specifically on these two tissues because they are the ones that are majorly used in traditional medicine practices. So now, to extract metabolites from these tissues, we used methanol as well as followed the hot water extraction method. Now, the second was done in order to mimic what is done in traditional medicine settings. As I mentioned, how these are used, the roots and the leaves of the plants would be boiled, and then the paste would be applied on the wounds and swellings to promote their healing. This was then followed by analysis of these extracts on liquid chromatography mass spectrometry platforms, and the generated data was then analyzed, processed using various platforms. Now, to annotate or put names to these chemical signals that we detect, we explored various computational tools such as molecular networking. This was then followed by biological interpretation. Okay, so I hope you can see. So making use of these various computational tools, we're able to tap into or annotate quite a lot of metabolites from the metabolomes of these two plants, from the leaves of the plants as well as the roots of the plants. Now, one trend or pattern that one can see is that we were able to annotate more of the flavonoids from the leaf samples in comparison to the root samples, while looking at the lipids, more of the lipids were annotated from the roots in comparison to the leaf sample. So this is just one way to illustrate how the use of these tools can actually help us tap into the chemistries or the underexplored chemistries of these traditionally used medicinal plants. Okay, so to put into context or show how exactly these tools help, so over here we made use of the tool referred to as molecular networking. Now, how this works is that it groups compounds that are similar in their spectral information or their fragmentation patterns together, and this same principle is then followed in the annotation step. What this means is that compounds that are similar in structure would be grouped together, and then how this helps is that if I'm able to identify at least one or two of these nodes, I can use that information to then extrapolate and postulate the identities of other unknowns in the same cluster. A typical example, so over here a node, the circle refers to a query spectra, which represents a metabolite of interest, and these two were matched to reference spectra in GNPS house libraries and putatively annotated as being the flavonoids nicotiflorin as well as rutin. So this is one way in which the use of computational tools such as molecular networking can help in terms of propagating metabolite annotation, reducing the re-isolation of the same compounds, as well as increasing then the probability of us discovering novel bioactive compounds that can be candidates for drug discovery. Okay, so another advantage of this tool, feature-based molecular networking, is that it allows us to visually represent the metabolome that we extracted. So you're able to visualize all the extracted metabolites that you got from your leaf or your root samples, and also it integrates or incorporates relative quantification information, helping us then to also see the differential distribution of different metabolites across the leaves of the two plants. So taking flavonoids for an example, we saw that chimephil retinoside, luteolin glucoside, and isoquercetin, as well as their related compounds, were more abundant or more predominant in the leaves of Melianthus comossus in comparison to the African potato. Whereas if you look at the flavonoids such as vicinine 2, corimboside, and saponin, these in this case were more predominant in the African potato leaves in comparison to the Melianthus comossus leaves. Now what this shows is that it speaks to the differences in the chemistries of the two plants that we are working with. Another interesting observation we made was that we annotated a lot of the glycosylated flavonoids. So to then zoom further into these, we created sub-networks of these glycosylation patterns. So over here, the shape of the node correlates to the type of flavonoid, while the colors of the nodes correlate to the sugar that is attached to these flavonoids. So we are able to see how there is quite an interesting glycosylation patterns of the flavonoids in these plants, and with the glucose conjugated flavonoids being the more abundant in this regard. We also looked at how these flavonoids were distributed across the leaves of the two plants, where we saw that chimephil in its A-glycan form, that is when it's not attached to a sugar, was more predominant in the leaves of Melianthus comossus, while when we looked at the glycosylated forms, we saw that chimephil 3 retinoside in this case was more predominant in African potato leaves, while chimephil 3 beta-xylose was more predominant in the leaves of Melianthus comossus. So this speaks to differences in the specialized metabolisms of the two plants under study, which can be translated to variations in how they are applied in traditional medicine practices. So this study, to conclude, this study contributes to global efforts to look back into medicinal plants as sources of natural products that we can use for the development of new drugs. And South Africa, with its rich biodiversity of the plants as well as the rich traditional medicine knowledge, stands to really give a lot in this regard. So this project is contributing towards efforts of also bridging the gap between traditional medicinal knowledge as well as modern science, and this can speak to policies, conservations, as well as strategies to implement some of these in the healthcare system. So from the generated maps, the use of the tools helped us to gain better insights with regarding to the phytochemistries of the two plants that we're working with. And we saw that they spend various chemical classes from tannins to flavonoids to ACA compounds, amongst many others. Moreover, we looked into the differential distribution of the metabolites across the leaves of the plant, speaking to differences in the chemistries of the plant. We also looked at the glycosylation patterns of these flavonoids and their distribution thereof, speaking to the specialized metabolisms of the plants that we are working with. And although not shown, another step that we did was perform computational modeling. So that is molecular docking as well as molecular dynamics simulations in order to predict the activities of selected metabolites. As part of the ongoing work, we are currently busy with the in vitro anti-inflammatory and anti-venom biological assays, and also exploring more of these computational tools to help us dig deeper into the chemistries of these two plants. So, yeah, this brings me to the conclusion of my presentation. These are my references. I'd like to acknowledge the following individuals and institutions and thank the CSIR for this opportunity to share my work. And last but not least, I'd like to acknowledge the Tukizimana Research Lab. Thank you.
Very interesting talk. Questions? Please go ahead.
Thank you, ma'am. Nice research and nice presentation. So let's say you had more resources like time, finances, and more hands to help you with your research. What would you have improved?
What I would have improved would be, okay, so I am limited with regards to the tools that I can use based on my limited coding and programming knowledge. So if I could get expertise in that regard, that will help me also now expand further. And yeah, and with the efforts towards the fractionation and the biological assays, that will really help us get to the more active fractions that we can then take further to NMR to structurally characterize the structures and identify these compounds that can be used as candidates then for drug discovery.
Thank you. Thank you very much.
Any more questions?
Thank you very much for the presentation. Very interesting. In terms of, maybe if you can go back to the slide where you are showing the processes, the analysis, the HPLC stuff.
The steps, the methodology?
Yeah.
Oh, okay. I'm going the wrong way. Sorry.
Oh, yes. So just looking at that, you mentioned that after your extraction and then you do your analysis on your LCMS and then the data mining, is there a specific software
that I use?
Yeah.
Yes, so depending on where you analyze, there are some vendor-specific platforms. For instance, with the Waters instrument, you'd use the Waters Connect software, but there's also open source platforms such as MZMine3 that helps us with processing these, and also it can even annotate as well for you as well. So yeah.
Okay, so will this be like sort of a foundation? Because if you go through your LCMS and then you have your data mining, will it be like a precondition for your isolation? So that you don't, because with your LCMS data, you wouldn't know what chemicals to isolate. So will that be like a shorter way to get to the type of compounds that you would isolate going further?
Yes and no. So in our case, we do not use it for that. So the first thing that we wanted to do was explore the phytochemistry or the metabolome in its entirety, regardless of what's bioactive and what's not. And from that, also using the in silico tools as well as from the assays would then narrow down our search to those that are specifically active against the venom and the,
Okay, thanks.
Sure.
Thank you for the presentation. I think my question, it might be related to us in regards to the data mining.
Okay.
I just want to know which techniques did you use, and also for your molecular networking,
Okay.
Now, how long does it actually take with your current tools?
Okay, so for the data mining and processing, like I said, I use the MZ Mine 3, so you can download it online. There's also MS Dial, which is another option that you can use. And for molecular networking, so it's an online-based tool called GNPS. So what you do is that after you process, you get two files. That's your quantitative information file as well as the one that has your spectral information. Then those are the files that you use to generate these networks. And with these, with the current updates with the tools, it actually takes very little time, in less than 30 minutes. Well, depending on how big your data set is, you can actually generate the network quickly.
So basically, is it like machine learning?
Yes, there's also some tools that incorporate machine learning and deep learning.
Okay.
Yeah.
In this case, which one did you actually use?
We use okay, so remember from that list of the many tools? So the results that I showed were from this one, which does not use machine learning. However, MS2 Query does incorporate some deep learning aspects.
Thank you very much.
So I'm going to take one last question. Go ahead, Devon.
Thank you so much for the presentation. During my presentation, I touched on the National Environmental Management Act. So I see your project has kind of like triggered that. How compliant is your project?
Yeah, to be quite honest. You know, when you presented, I was like, I hope she's not going to ask me that. Yeah, I'm actually not sure, but that is something to look into. We did not go outside to the traditional healers as well to try and get information from them. But we can speak further.
Yeah. Even the research itself, it triggers the act.
Oh, yes, because you can't just like take a plant and do some analysis on it. Yes, I do understand.
Okay, thank you.
So that leads us to the conclusion of the session. I would like to sincerely thank our speakers and all of you for participating and making it a really nice and engaging session. And yeah, can we just give all the speakers the last round of applause? And then please feel free now to go and have some lunch.
My name is Yonela Tukwayo. I'm a senior researcher in the Energy Center. I am an economics by background. What that means for the speakers is that when it comes to the title of your presentations, you're gonna have to say those yourselves. So presenters, you have 15 minutes for your session to do your presentation. And then in the last 5 minutes, we're gonna do questions and answers. When we have 2 minutes left to your presentation, I'll start making my way slowly to the stage. That simply means that you must start wrapping up your session. In the last 5 minutes, we're gonna have a Q&A. For that, I ask colleagues that we please keep our questions brief, as well as our responses brief, so that we can get home as soon as possible. So with that said, I would like to introduce our first presenter, Tabeni Langa. ICT, please help us.
Good afternoon, everyone. My name is Tabeni Langa. I am a researcher in the Bioprocess Development Group. Today, I'll be sharing the project that I worked on in the past 2 years, titled Fungal Mycelium as a Sustainable Protein Source from Development to Pilot Scale Production. As we all know, protein form an important part of the human diet as it supports general body functions in terms of degrading the food that we eat and also provide storage molecules and also assist in synthesizing the hormones that our body needs, as well as supporting our skeletal movements and general immune responses of our body. And having a balanced protein diet generally assists in maintaining a healthy body weight. Here on the right-hand side, I had an example of the foods that we take in our daily diet that provides us with a balanced protein diet, which constitutes animal and plant-based protein sources. However, conventional protein production systems are associated with various challenges that have been reported globally, which means that the current systems do not have the full capacity to provide for the demand that is required, with the rise in population that is estimated to be 10 billion people by the year 2050, and the general global shift in health awareness, and the fact that overconsumption of meat is associated with causing non-communicable diseases, which ended up in shifting consumer preferences. And also with plant-based production systems, there's reported agricultural instability that is caused by climate change issues. And lastly, the livestock production is also associated with negative environmental impacts, such as emission of greenhouse gases. Fungi has been used as a sustainable protein source for over 1,000 years. This includes edible mushrooms that are well known that we have in the market, including button mushroom, shiitake, oyster, as well as enoki. And as of 2002, there was a vegan protein alternative that was introduced that was referred to as, or it is provided by corn, and it is produced through a solely liquid mycelium production process. And in general, fungi provides high nutritional food products with low calories and low fat that are high in fiber, rich in antioxidant, and a protein content ranging between 19 to 55%. In South Africa, the mushroom production industry is dominated by Agaricus bisporus, which is button mushroom, and other species, Pleurotus austratus, as well as Lentionella edodes, are also at the development stages. However, there are various challenges that are being experienced by this industry as these food products take longer to grow. There are high operational costs involved, and the biotechnology strategies, which is mostly through cultivation in liquid mycelium, there is a lack of the infrastructure there in the industry, as well as the skill gaps and the lack of post-harvest infrastructure as these products are highly perishable. Therefore, the CSIR saw opportunities in assisting or developing these technologies, which then constitutes one of our values in localizing commercially relevant technologies, thus contributing to the sustainability development goals in terms of addressing food security issues, being able to implement circular bioeconomy business models, and thus influencing the socio-economic status of our country. Generally, mushrooms are saprophytic fungi as they can grow on dead trees in the forest, and for commercial purposes, they are produced through a process called solid substrate fermentation through use of either grain spawn or liquid spawn, which then feeds to the mushroom solid substrate fermentation. The liquid spawn is known to provide faster growth rates compared to the grain spawn, so it is one of the main biotechnology strategies in addressing the issues that I addressed earlier. And then for mycoprotein, it is mainly produced through liquid submerged fermentation. So then the aim of the study was to conduct development and demonstration of cultivation processes of fungal mycelium for use as sustainable sources of protein. These were the objectives were to select nutritionally and commercially relevant fungal strains with the potential to be used as sustainable protein sources, investigate their cultivability, and to develop scalable production processes up to 30-liter bioreactor scale, and to demonstrate the production technology of the best performing strain at pilot scale to validate the commercial viability of the developed technology, and to lastly assess the performance of the produced biomass in terms of yield, protein content, as well as nutritional profile. This is just a summary of the process of the R&D approach that was taken because to make sure that we get to repeat the results that we obtained at the end of each process, we needed to make sure that each of the starter culture that we have throughout all the research batches that we needed to perform, we have a reliable culture stock. Therefore, we needed to isolate a pure culture of each isolate. We needed to establish reliable and reproducible cell banks. And then we needed to assess the growth of each strain at flood scale to make sure that by the time we move to 30-liter bioreactor cultivations, we have at least established the medium that we're going to use and the growth conditions, which was then optimized as the scale to ensure that we achieve reduced process times. And the developed technology was then moved on to 1,000-liter scale production, which is pilot scale, and we developed different solid liquid separation processes. As in the bioreactor, we would obtain a culture that is mixed with spent nutrients, so we needed to separate what we produce and make sure that the product that we get is a product that we can assess the nutritional content and make sure that it looks like what a mushroom would look like. For establishment of our cell banks, because of the nature of the strains that we were working with, we had to determine to use two different techniques that would be suitable for each strains that we were working with. For instance, for so for all 4 strains that we were working with, we tried 2 different techniques, namely agar discs, cell banks, as well as liquid mycelium. For agar discs, we noticed that it was mostly suitable for mushroom strains, as there was a homogenization step that was required before starting the process for these strains. However, this technique was time-consuming and had higher risk of contamination, which is what we do not want for our process, as we needed to have a pure starter culture for each and every process. And mushrooms are very susceptible to contamination. And then luckily for Physarum venenatum, we managed to see equal reproducibility when both techniques were used. So these are just images of our cultures when we were validating the reproducibility of our cell banks, and they did show to work. Okay, so in terms of demonstrating the cultivability of our strains, firstly, I would like to mention that we observed in the previous step that the Agaricus bisporus strain was still taking longer to grow, which it was about more than 30 days. So for the sake of time, because we only had a year to complete the project, so it was not suitable for that particular time, so we removed it and we moved on with the 3 strains, which is Lentinella edodes, Pleurotus ostratus, as well as Fusarium venenatum. We selected different medium formulations. For some strains, it ended up having to be about 11 different recipes, but for the sake of this presentation, we selected top 3 that showed to give us acceptable performance. I will start with the Lentinella edodes. For Lentinella edodes, we saw acceptable growth of about above 2 grams per liter of biomass in the medium designated as test 3 in the presentation. And for Pleurotus ostratus, it was about above 18 grams per liter in the media designated as test 2. And for Fusarium venenatum, we got about 29 grams per liter in the medium designated as test 3. All these medium, the differentiating factors were different concentrations of the carbon and nitrogen sources, and they demonstrated variation in their growth rates, with Fusarium venenatum growing within 2 to 3 days, which was suitable for our process and the objectives that we had set. We moved on with the condition that we had developed at flask scale, and 2 30-liter bioreactors with Lentinella edodes. We still observed similar performance that we saw in flask cultivations, and with Pleurotus ostratus and Fusarium venenatum, we did achieve productivities that were comparable in literature. However, I'd like to point out this biomass yield here. It was mainly due to the fact that the concentration of the sugar that was added in the beginning of the process was a bit lower compared to the Fusarium venenatum. However, the results that we got were comparable to what has been reported in literature in about 3 days. As you can see, most people cultivate these strains for a bit longer, even in bioreactors. Because of resources, obviously, we were not able to go longer than 3 days. And for Fusarium venenatum as well, we observed mostly similar results with 21 grams per liter of biomass that we obtained in 44 hours. For development of our downstream processes, because of the nature of the broth that we would result after the cultivation process, we would recover about 11% of our biomass, which does not mean it's low because of the nature of the mushroom. That's the only maximum yield that we were able to achieve. And then we tested different drying techniques, with freeze drying being best suitable for the Pleurotus ostratus. However, we observed higher losses of the product when this technique was used. And similarly, with the Fusarium venenatum, we got about 11%. And for this one, we tried both freeze drying as well as tray drying. However, we saw that the freeze drying, the product maintained the nature, the similar appearance as the wet biomass, whereas the oven drying changed the appearance of the product, which is also attributed to the different techniques or different processes that these techniques afford drying. The freeze drying uses the drying process, the ice and water molecules are removed from the cells, whereas the oven drying applies heat to the biomass, hence the change in the product appearance. It was mostly said that the technique that can be selected should be determined by the operational cost, with oven drying being most cost effective and freeze drying a bit expensive. So we also moved on to demonstrating our process at 1,000-liter scale, and this process required a two-stage step, as we needed to start at flat scale, move on to 30 liters to get a seed culture of about 30 liters to transfer to the 1,000-liter cultivation vessel. We did optimize growth conditions here, and the seed was harvested after 23 hours of cultivation. And in the 1,000-liter, we observed a similar performance with the feeding of the bioreactor started after 24 hours. As you can see, we harvested the seed at 23 hours, and when we got to this production vessel, the feeding, which means after the depletion of the carbon source, we needed to add glucose to the vessel. Because also of the nature of the biomass that was being produced, we were not really able to maintain the dissolved oxygen. However, that is in the process of being optimized. And this is the nutritional profile of the product prototype that we generated that were comparable with the commercial benchmark, which is corn, and even the freeze-dried biomass showing a little bit high protein content. In conclusion, we have successfully developed the liquid mycelium production processes and demonstrated the technology at pilot scale, which showcases the potential or capability to localize this technology in South Africa. The growth challenges for other two strains are observed in literature, and further development strategies are required. However, there is a potential application for these in terms of providing the local producers with the liquid spawn, because the grain spawn is still contributing to the longer growth cycles that are observed. And we also recommend the use of models to optimize growth conditions. And the current work is to optimize growth conditions at production scale with potential use of waste streams to apply the circular bioeconomy business models. And the project is one of these will be implemented in the current phase of the project, which is for commercialization under the C-Cubed enterprise. I would like to acknowledge my supervisors, and thank you.
Thank you, Tabeni. Well done. Thank you for that very interesting presentation. Colleagues, do we have any questions for Tabeni?
Hi, I'm just curious to know, doesn't the process of either freeze drying and oven drying interfere with the protein capability of the product? Because I would assume like there is denaturing that you're trying to avoid by all means.
Okay. We saw that the freeze drying did not have any negative impacts, but with oven drying, we did see that it had lower protein content compared to the same product that was freeze dried. So the recommendation that I think we have is playing around with the temperature and making sure that at least we dry at about 30 degrees, because it does dry, it's just that it takes a bit longer. Yes.
Thank you, Tabeni. I saw you mentioned your yield, biomass yield, 11%. Does that correlate to your protein recovery? How much protein did you recover?
No, that's not the protein. It's like the physical biomass that we extracted from the broth that after fermentation. It's just the dry, the physical biomass.
So did you measure the
protein
protein recovery? No?
Not yet. Oh,
Okay, okay, because I was just going to say that maybe once you've done that, you can compare to other sources of protein to see the performance.
Okay, we'll do
Thanks.
that. Thank
Sharp.
you.
Thank you, Tebeni. Well done. Our next presenter will be Brittany Cedris.
If you can just use a round of applause.
Good afternoon. I am Brittany Cedris from the University of the Free State, and my project focuses on valorization of biomass, particularly investigating the enzymatic breakdown of biomass to produce complex oligosaccharides. At the center of the circular economy, lignocellulosic biomass presents itself as a particularly renewable and underutilized product. It composes of three major components: hemicelluloses, celluloses, and lignin. Hemicelluloses are particularly noteworthy for their abundance in xylem. Xylem acts as a precursor for xylo-oligosaccharides. These xylo-oligosaccharides are known to have antioxidant properties which are linked to their side chain substituents. The challenge, however, is the recalcitrance of lignocellulose, which is difficult to break down. This is due to the intricate cross-linking between the polymers. Enzymatic hydrolysis provides an eco-friendly, resource-efficient solution to break down this biomass. My study aims to characterize the C1184 secretome from A. spigelus niger. My study aims to characterize the C1184 secretome from A. spigelus niger, produce and quantify complex xylo-oligosaccharides, and look at their antioxidant properties. For substrate specificity, we examined the C1184 secretome with 1% weight per volume substrates for glucans, xylins, as well as starch, using 50 millimole sodium citrate buffer pH 5. We did the reactions for 1 hour at 37 degrees Celsius. For biochemical characterization of the C1184 secretome, we tested the pHs for a pH range between 4 and 9, temperatures between 30 and 50 degrees Celsius, and the thermostability was tested for hours 1, 5, and 24 hours for temperatures 37, 50, and 70 degrees Celsius. For kinetic parameters, we used the mechanismentum for substrate concentrations ranging from 0.05 to 16 milligrams per mil. Zymogram analysis was done to look at the specific enzymes involved in hydrolyzing this biomass. We did a native page for cellulase, xylin, galactomannan, and starch. The oligosaccharides were produced by doing a 24-hour incubation with the C118 secretome and beetroot xylem. We then did precipitation using organosolvent using a 1:4 ratio solvent solution with ethanol. This was then, the resulting pellet was then dried and resubmerged in sodium citrate pH 5 buffer, where we then did LCMS analysis. We also used this to do further reactions using esterases, so 2 fural esterases and glucuronol esterase and a GH10 xylanase. This was analyzed using TLC as well as looking at their antioxidant quenching abilities using 2 common methods, ABTS scavenging as well as H2O2 scavenging. For substrate specificity, we saw that the enzyme has multiple activity across xylem, cellulase, and starch substrates, with xylem being higher. This demonstrates that the secretome is a multifunctional enzyme. This was further demonstrated by the zymogram analysis, which shows us multiple bands for the various substrates. We see multiple band profiles on cellulase, xylem, starch. However, for galactomannan, we did not see any activity. For the characterization of the enzyme worked optimally at pH 5. The optimal temperature was 50 degrees Celsius. However, thermostability revealed that the enzyme remains stable over time at 37 degrees Celsius. The kinetic parameters revealed that for starch, the Km being low shows that the enzyme is able to hydrolyze the substrate even under low concentrations. For xylem, we see that the catalytic constant but the Km value is high. So the enzyme is sufficiently able to hydrolyze the enzyme, however, not when the enzyme is not abundant. For the oligosaccharides, using LCMS, we had the major product produced being xylotriose, followed by xylobiose. With the second hydrolysis with the C118, removing the side chain substituents, we see that the enzymatic profile or the TLC profiles are different compared to that of the C118 secretome by itself. The control we see at the baseline, the substrate remaining at the baseline. With the glucuronol esterase and the fural esterase, we have a very distinct profile, with glucuronol esterase having a very defined band close to the baseline and it having very faint bands towards the end of the plate. Similarly with fural esterases, xylanase, being a GH10 enzyme, has many defined bands across the TLC plates. For antioxidant, for A, we look at the green bars, which present the H2O2 concentrations, the control being the H2O2 control, showing the total H2O2 radicals present. The blue line presents the antioxidant radical scavenging activity of the various oligomers with the side chain enzymes. For A, we see that with the fural esterases as well as the glucuronol esterases, we have more scavenging abilities. With B, the blue bar shows antioxidant garlic acid equivalents, and the green line showing antioxidant scavenging. We also then here see that fural esterases scavenge the unstable radical more. This is, according to literature, we know that the antioxidant properties of oligosaccharides are linked to those extra, those bonds formed between those double bonds with ferryl esterases and the extra bonds that can form sugar conjugates with the radicals. In conclusion, we have successfully characterized the C1184 secretome, looking at its zymogram, looking at its xylanase, cellulase, and amylase profile, enzyme profile. We also then looked at the biotechnological promise of producing oligomers with antioxidant properties, and this aligns with the United Nations goal of Sustainable Development 12. Thank you. I'd like to acknowledge the following people.
Thank you, Brittany. Well done on all your hard work. Any questions for Brittany?
Okay.
Thank you, ma'am, for such a wonderful presentation and the good research that you've done in terms of characterizing starch and proteins. So within these characterizations that you were doing, is there anything you would consider not so obvious that you did?
So with this study, the secretome, the C1184 secretome, was actually purchased as a cellulase from Sigma. And so when we characterized, we saw that it had significant activity on xylane and starch substrates. So not what you would see with a cellulase, and the activity on xylane being more than that of cellulase kind of started giving us the idea that perhaps it's a secretome and not a cellulase.
So within those discoveries, did it inform you into maybe developing a better characterization framework?
Not necessarily. Something that we are currently looking into in our lab is molecular docking, which would then be more resource efficient. So yeah, that's one thing.
No, thank you. Thank you very much.
Okay.
Thank you for such a beautiful presentation. I just have some few questions with regards to your kinetic parameters, especially on you mentioned Vmax, okay? Which is your maximum
velocity.
Exactly, yeah. How is your Vmax determined? Was it determined experimentally for your enzymes?
I did it by using a range of concentrations for my substrate, and then we did it using the Michaelis-Menten formula.
Formula, okay.
Yes, and that we then used Excel Solver X to then plot the graph and then compare it based on the Michaelis-Menten formula.
So on the graph, okay, no, I get it. Thank you.
Okay.
I don't know if I missed your source of lignocellulosic material.
So we used a range of substrates because the first objective is to characterize the enzyme, so therefore looking at the different enzymes involved. So we used commercial substrates since it's an honors project. So we used a substrate from xylane, cellulase, and as well as starch.
Oh.
So they were all purchased from Megazyme and Sigma.
Okay, thanks.
Okay, thanks.
Colleagues, our next presenter is Machera Letswalo.
everyone. I thought when they said this is a revered session, all the nerves also die off. So as introduced, my name is Machira Parsons Litswalo. I'm the country researcher here at the CSIR. I'm from the Bioprocessing Development Group. The talk today is going to be about the large-scale production and application of biocoating product for the fruits and vegetable industry. I'm going to give you an overview of how we developed our product and also how we applied through field trials. To give a bit of a background, large-scale commercial agriculture, especially horticulture, which is the production of fruits and vegetables, is a major part of our economy. It contributes to our country's GDP and is a primary source of foreign exchange through exports. This industry currently is moving towards the use of biocoating agents as opposed to chemical waxes. This is because biocoating agents are susceptible, are sustainable, and are an innovative approach to managing our fresh produce after they've been harvested. They are edible, they are non-toxic, and they provide a safe way to protect our food. So one might ask, what are biocoating agents? These are materials used to create a film or a thin layer, a thin protective layer on the surface of fruits and vegetables. They are made from natural and food-grade ingredients, such as the lipids, polysaccharides, which are your sugars. To give a bit of a context, post-harvest degradation of fresh produce is a global issue because fruits and vegetables are susceptible to natural processes such as microbial contamination and also moisture loss. The scale of the problem is bigger than we think. According to reports, 30% of our fresh produce is lost between the farm, the market, and the consumer, which now poses a threat to our food security. The problem with the current use of chemicals by farmers raises health and environmental concerns. When we looked at these challenges, we saw that there's an urgent need for a sustainable and safe, effective approach or product, especially for the export market. So in this project, we aimed to develop a scalable bio-based preservation technology. And to achieve this, we set out two objectives: to demonstrate a scalable manufacturing process for the production of our biocoating agent, and also to validate the product's efficacy through field trials. And the expectation out of this project was to see a reduce in reliance on these chemical waxes and pesticides. Most importantly, to extend the shelf life of the produce, which will help improve the distribution internationally and nationally. So one might ask, how do we then produce this biocoating? If you put your focus on figure 1, this is a process flow sheet of how we produce our biocoating agent. So we start by inoculating a bacteria, a microorganism, in a flask. We then transfer the microorganism into a 300-liter bioreactor, which is under controlled conditions, to produce our EPS. Those are sugars. After fermentation, we harvest and centrifuge to separate the exopolysaccharides from the microbial cells. We then mix the exopolysaccharides with food-grade ingredients. Just that based on IPs, I cannot give mention to say which ones are those. We then dry the formulate, we mill it into a fine powder, and then we have a ready-to-use product. The product met all required quality and safety specifications, which allowed us to then continue with our field farm trials. The trials were conducted in collaboration with the farmers in South Africa. Three produce, which are grapefruits, tomatoes, and oranges. The study was conducted on these three fresh produce. For the grapefruit, the study was conducted for about 90 days at a Solil farm in Hoedspruit. This is where, come back to the slide. This is where, if you put your focus on figure 3, this is where you see the schematic diagram shows you the final product, as I mentioned, is a powdered product. This is where it was mixed with water and spray coated onto the fruits, which were then dried and sorted so that we eliminate those ones that are already compromised for the purpose of our study. They were then packaged and brought to us at the CSIR for us to do our further studies. Go back. So if you focus on figure 2, it will show you that the CSIR, those produce, there were two studies that were conducted on the food produce. One was applied with the CSIR biocoating agent. The other one was coated with citrosol, the citrosol from the farmer. From the diagram, you can see that the CSIR biocoating demonstrated better moisture retention compared to the commercial benchmark. Grapefruits treated with the CSIR product showed moisture loss of 21.86%, whereas the citrosol treatment batch had a 47% loss. The qualitative analysis showed the CSIR coated fruit maintained better texture and had delayed decay compared to the control. So upon our analysis, we realized that in as much as it wasn't a microbial inhibition, we realized that somehow there are pathogenic organisms that are growing on our produce. However, as I said, the CSIR biocoating agent was able to delay that decay. So, trial 2 was on the tomatoes, where three batches were assessed. One was coated with the CSIR biocoating agent. The second one was coated with the farmer's chemical of choice, which was sodium metabisulfate, and the third one was left untreated. From our analysis, the CSIR biocoating resulted in a lower mass loss compared to the untreated and those treated with sodium metabisulfate. By day 30, the fruits coated with the CSIR biocoating agent showed a mass loss of 9.33% compared to 10 and 9.8% of the untreated and the sodium metabisulfate respectively. This shows that the CSIR product effectively delays wrinkling and softening. This can be viewed on figure 5, where you can see on image B, you can see the wrinkling. I don't know if it's visible. The third trial was done on oranges, where 2 batches were analyzed. One was treated with the CSIR biocoating agent. The other one was treated with the citrosol, the same one that was treated with the grapefruits. So in this case, both the products didn't show any much long-term efficacy. However, from what you can see on the pictures on picture C and D, those oranges were treated with the citrosol. The pictures in A and B were treated with the CSIR biocoating agent. It shows you that the CSIR biocoating agent was able to in as much as it didn't assist with reducing the moisture loss, but it assisted with cells, microorganisms, pathogenic microorganisms growing on the organisms, on the fruits. In conclusion, we can say that we managed to demonstrate a scalable end-to-end manufacturing process of a product that is safe, biodegradable, and it's effective in prolonging the shelf life of various fruits by reducing water loss and microbial decay. The technology it's also sustainable. It's a sustainable alternative to conventional chemical preservatives for agricultural sector. In our future work, we are looking to conduct other large-scale trials across different seasons and on other produce like mango, pineapples, and grapes. We are also looking to perform toxicological and safety assessment to meet food safety regulations. We are also looking to explore combining the CSIR biocoating product with other preservative methods. With that said, I would like to thank you and also acknowledge my supervisors.
Well done, Mashere, on a very excellent presentation. Any questions for him? Okay, we'll start with the gentleman at the back with the white shirt.
Yeah, thank you for a very good presentation. What I'm interested to know is the effect of different weather conditions on it. So if you had the coating on a tomato, for instance, when I saw in your second trial, if it was kept in a cold environment versus in a very hot environment, how would that affect the mass loss?
Currently, I think that is the second stage of when I said in the next phase of our research is to look into including other preservation methods. That's when we'll go into temperature to see if in multiple temperatures, where is it best to. We haven't got to that study yet.
All right, perfect, thank you.
I'm not the one who's calling.
Okay, thank you. Actually asked one of the questions I wanted to ask, but I've got, I think, two more. I saw that you performed some benchmarking with, standard, methods. So I want to ask why only one? Is that the best in the market?
In terms of the citrus oil, because it was tested on oranges and also on grapefruits, which are from the citrus families. We saw that from the farms that we got the fresh produce from is what they were using to preserve their fruits.
Okay, so
that's why
you got it from the farm.
Yes.
Okay, but did you go further to investigate what other people use so that you can at least be able to benchmark your product with the current standard with other product? That's one. But like you said, you can introduce that in the future. Yeah, thank you. The second question is the production cost, which we actually also influence the selling price.
Yes.
So how does your production cost compare to the existing ones? And
currently, I would say because we are running it at a 200-liter scale, because the major, in terms of techno-economics, you can see that the major cost comes from production, which is when we are running the 200-liter bioreactor. But because we have a system, a software, Super Pro, that is what we can use we used it to simulate, to say, if we take it to 1,000-liter, what happens? And we realized that in most manufacturing processes, when you upscale, you then reduce the cost. At small production, then I could say, the production the cost is a lot. But when we upscale, then we take down the cost.
Okay, sorry, thank you for that answer. So my last question is similar to what I had. Okay, thank you.
I'm more interested in the mechanism of action of the biocoating product. So, what component affords it the ability to maintain the quality of the produce, and what component could potentially afford it the quality of being antimicrobial?
From from research, I think one of the presenters had spoken about exopolysaccharides, them being able to inhibit the growth of other microorganisms. So this is from a study to say exopolysaccharides are capable of inhibiting growth of other microorganisms. So because we had a microorganism in-house that can produce exopolysaccharides, then we thought it fit for us to try and see if we can find a solution for the agricultural sector.
So this means basically with this biocoating, which is based on the EPS, your target is actually microbes, like you're hindering microbes in order to further to allow the fruit or vegetable to maintain.
Yes.
Oh, okay, got you.
I think just one last one. Ronald, because I just see the CSIR byproduct agent. Do you care to share what's that? Or is it,
I did
or is
mention
that an
it's
IP kind of
yeah,
issue?
it's a position that I cannot. But there's, let's see. Yeah, since you see there's starch in there, maybe you can start from there. I just gave you 25%. I gave you 25%.
All right, no. All right, well, thanks, man. I just wanted to.
Thanks, man, for the great presentation. I just have one question with the precursor. The precursor is, at what TRL level is this product?
TRL level 6.
6,
Yes.
okay. So I think we are all aware of the advantages of products like fertilizers, and also some of the disadvantages that they tend to have upon consumption. So I want to understand if whether at this point in time, have you guys already looked into studies that look at the disadvantages of overconsumption of this coating product, given that you said it's all... Sorry?
Second stage.
Oh,
that will
that's
be in our second stage.
Okay.
How would it affect you when you consume this much of the product? That is will be in our second stage. Yes.
Thank you again for the opportunity. Thank you for the presentation again. So my question is, following your experiment, I want to know what storage conditions did you use? Because you didn't tell us what storage conditions. And then that condition you use, is it the same with what the farmers use with the product you compared it with? Because if the conditions are not the same, it might affect your results.
Oh, yeah. No, we set out them to say, okay, what temperatures do you store your fresh produce before you export or you distribute? 19 degrees Celsius. So it was good that our labs are always at 19. So we just had to get a lab so that we can store them there. Yeah.
Thank you, colleagues. For the next presenters, colleagues, I was kidding when I said we need to get home quickly. Take your full 15 minutes of fame. We are all here to listen to you. So with that said, our next presenter is Stefan Karamaski.
All right. Good afternoon, everybody. Today I'll be presenting on the topic using battery energy storage systems and renewable energy to reduce municipality costs, a South African town case study. So just a little bit about me. My name is Stefan Karamanski. I have a background in electrical and electronic engineering, and I've also completed my master's in renewable energy engineering, and I work as a researcher here at CSIR within our energy center. And I promise you, I don't only own CSIR branded t-shirts and shirts. I've also had the pleasure of being on site a few times, and it's really been a positive experience. So these are just some of my focus areas, including wind energy, solar PV, hydrogen, smart grids, etc. Okay, so this is just a little bit about what we'll be talking about today. So I'm sure you're all well aware that South Africa is in the midst of an energy crisis. However, this has also sparked some kind of development and innovation within our country. Embedded generation is on the rise, and with renewable energy penetration increasing, we've seen an uptake in commercial and residential sectors. Municipalities have also noticed the advantages of renewable energy, and the City of Cape Town has used hydro pump storage to reduce the levels of load shedding within the city, and this has improved the quality of life and service delivery, as well as reduced economic losses and traffic congestion. So in this specific case study, we're going to look at a hypothetical scenario in the town of Hermanus, which you can see with the yellow pin on the right over there. And this Hermanus municipality is interested in a hybrid approach, including wind, solar PV, and battery energy storage systems, with a minimum 50% renewable energy penetration, and this is to meet the town's sustainable development goals. They are also interested in electric vehicle charging facilities to keep up with the latest technology, and this is quite an interesting case study because of the specific tariff structure of the Hermanus municipality. So I'm sure we've heard this story churned over and over again about load shedding, but I thought this is just quite a nice visual of load shedding over time, and we can see that we were hit pretty hard in 2023 with a lot of load shedding, and just when we thought we could get out of it in 2025, it came back to bite us. But what's most important about this is that we should be well aware that there were many times when we were on the brink of having load shedding, and it's not something that we should just dismiss for the future. So Eskom has also published data of rooftop PV. This data is a little bit old, coming from 2023, but you can see that there's a significant increase, even month on month, of the estimated installation of rooftop PV in South Africa. And as of September 2024, Eskom estimated 6.1 gigawatts of installed capacity of solar PV, which is a very, very big number. We've also seen a big reduction in renewable energy tariffs. So on screen now you can see the average tariff cost from bid window 1 to bid window 5, and we can see a reduction of 78% and 91% for wind and solar respectively from bid window 1 to bid window 5. And this picture doesn't tell the whole story. In actual fact, in bid window 5, the lowest bid was 34 cents per kilowatt hour, which is a very competitive price for Dwarsrug wind facility. So not only have we seen a reduction in renewable energy costs, but we've also seen a reduction in lithium-ion battery costs. Here you see a plot over time of the reduction in cost of lithium-ion battery packs, which you can see as an exponential decrease. And with variable renewable energies being intrinsically inconsistent, batteries assist us in creating a smooth, reliable power supply. And this results in the combination of wind, solar PV, and battery energy storage, creating an appealing solution to many of our energy supply problems. So lithium-ion batteries also have the added benefit of being able to reduce costs by peak shaving, time shifting, and arbitraging, while supporting the self-consumption of renewable energy. So this is just some background on load profiles. This is a study I did in 2023, looking at the South African wind farms diurnal profile, which is just a fancy way of saying a 24-hour-long period. So we can see there a red and a blue plots. The blue one is a typical power from the 36 operational wind farms in South Africa, and on a twin axis you can see the Eskom demand. And we can see that they align quite closely with the peak time, and this also creates opportunity for solar PV to come in and fill that gap during the day hours. So just remember this demand profile, the Eskom demand profile, for later. So we have the technology, we have the natural resources in South Africa, but what about the grid connection? And this is a big problem for those of you who know about the challenges about renewable energy in South Africa. In this study, we assume that there is sufficient grid connection. despite the Western Cape, Eastern Cape, Northern Cape, and hydro regions being completely saturated at this point. So electric vehicles have also seen quite a large uptake in South Africa, and this data represents that. We've seen a 67% change from 2022 to 2023, and 110% change from 2023 to 2024. And this is expected to rise for the next year. And if you've even been paying close attention while driving around on the roads, maybe you might have even seen the latest Toyota hybrid, or maybe a new Chinese EV on the road, or maybe you've even seen some of the EVs here on the CSIR campus. So this is no longer just a thought, it's actually a reality. So moving on to the methodology now, Homer grid was used for all of the modeling in the study. And the demand profile of the Hermanus municipality is not necessarily something that is easy to access or publicly available. So remember that demand profile I showed you earlier from Eskom? That is just a representation on screen now of that normalized profile. And that profile is then scaled to the population of Hermanus using census data, and then takes into account the population growth rates for the subsequent years. So we want our system to not just provide power, but we want our system to provide power reliably. And I mentioned to you about the risks of load shedding, so we also incorporate grid outages into the model, and 100 2-hour sessions are incorporated. So for each one of those black stripes you see there, that is when the grid is not accessible for any power delivery. From literature, an 87 Rand per kilowatt hour penalty is applied to incorporate any economic losses if energy cannot be served. So as I told you before, the Hermanus municipality has quite an interesting tariff structure, and as they form part of the Overstrand region, their time of use rates are used. And you can see here they have a low season rate, they have a high season rate, they have off-peak standard and peak times, and these are all incorporated into the model. The import and export rates in the model are used as the exact same figures. However, this is not really realistic because typically the export rate is a lot lower than the import rate. So in other words, you're selling back to the grid at a much lower rate than you are importing it. A 47 Rand per kVA demand charge is also incorporated, and this is to penalize users for high demand, which we will show how renewable energies can solve this. So in other words, you're going to pay for every amount of power that you receive, you're going to pay for this. So in other words, you want to minimize that. So in terms of the energy storage, lithium-ion battery energy storage is used, and the reason for this is simply because it can store large capacities, has a fast response, is able to support variable renewable energy sources, is scalable to large systems, and has improved significantly in safety and cost, as I showed you before. The Homer Advanced Storage Battery Model is used with an efficiency of 92%, with a minimum state of charge, or SOC, of 20%. So in other words, we only charge the battery to a minimum of 20%. We can't discharge it further than that, and that is in line with most manufacturers' recommendations and requirements. The degradation limit is set to 30%. So in other words, if the battery degrades to 30% of what its original capacity was, the battery is deemed no longer usable. And it's not wise to size the battery energy storage system based on peak demand, especially in this scenario, because the peak demand occurs for such a short period of time that it's quite easy to oversize the system. So instead, we look at the maximum of the average load, and that is 6.8 megawatts. So if you take that into account, the fact that two 2-hour load shedding sessions can occur one after the other, the SOC limit, as I just mentioned, to 20%, adding some capacity for arbitraging, and then we end up with a battery energy storage system of 39 megawatt hours. Okay, so in terms of energy generation, we mentioned before that there's this requirement of a minimum of 50% renewable energy penetration. So we have obtained solar and wind resource data over the past 22 years to look at the long-term climate of the region. The wind and solar energy are used in tandem, more specifically 6 megawatts of PV, 4.5 megawatts of wind. So in total, we end up with 10.5 megawatts of renewable energy. More specifically about the PV, in the model, the Homer generic flat plate module is used, and the Homer 1.5 generic megawatt wind turbine with 80 meter hub height. So we also included a 25 megawatt converter with zero cost and 100% efficiency. And the reason for this is simply just to not try and intervene with the renewable energy results, to try and keep those results separate to the influence of the converter cost. So I just want to re-emphasize that the reliance on the grid is not completely removed, so we can still make purchases from the grid, but we're just combining that with our renewable energy sources. Okay, so in terms of the EV charging, here you see a yearly profile. I mean, you can see where it's highlighted sort of in the middle from about 6 a.m. till 7 p.m. Basically, the day hours is where we're having the most charging as expected. And we have incorporated 7 high-quality 150 kilowatt chargers with Homer Grid's highway charger load profile with a 20 minutes average session and assumed 40 sessions per day. And we have a queuing system which is implemented, so in other words, if all of the chargers are being used at any one time, there's a queue that is created, and as soon as one of the chargers are available, it is allocated to a new user. The split for large and small EVs is 30 to 70%, 30-70, and the reason for this is we anticipate that there'll be an uptake of small EVs much more significantly than large EVs simply because of the high cost of EVs. Okay, so it's not really too critical to understand what's going on over here with the economic parameters. These are just all incorporated into the model. Maybe something of interest here is that the project lifetime is 15 years, but all of these values are taken into account in the economic modeling. That's just there for completeness sake. So moving on to the results. Hopefully nobody's fallen asleep by this point because this is the slide that hits home. So the design system has an internal rate of return of 25.3% and a return on investment of 39.5%, as well as a payback period of 5.1 years. And the system does indeed achieve the requirement of 50% renewable energy penetration, achieving a percentage of 53.9%. There's 0 kilowatt hours of unmet load, and the PV and wind capacity factors are 16 and 44.1% respectively. And the reason for this very high wind capacity factor is because Hermanus is quite close to the ocean and benefits from the low surface roughness of the ocean, so it generates quite high wind speeds in that region. The EV charging stations are also implemented successfully with 14,500 sessions per year with no unserved sessions. So most importantly, the annual consumption and demand charges savings are about 173 million and 2 million rand respectively, which totals a total utility saving of about 175 million rand. Okay, so this is just an interesting snapshot of the system. It might be a bit small, but I will guide you through it. So as you can see on the top plot over there, the red is the load, so that's the load of the municipality, how much energy they're using at any given time. The blue is the battery state of charge. So in other words, when it's saturated at the top there, it's at 100%, and you can also see at the bottom, it never goes below the 20% rule that we set out before. The yellow is the grid demand charge, and whenever there's a dip in the grid demand, that just means that there's currently load shedding happening right now. In the bottom plot, the orange is the grid purchases. The green is the solar PV output, and you can actually see the tracking throughout the day when the sun rises and peaks. You can see that the solar PV tracks that, and then the wind, which is quite erratic in nature as per expectations. So one interesting thing is that if you look over there, sort of in the middle of the screen, we can see that there's an instance of load shedding, and we can see that there is currently a demand indicated with this red. And because the blue and green insufficiently are able to supply that load, and we have no grid purchases because there's load shedding, we can actually see a dip in the battery states of charge. So the battery, in other words, is supplementing the load. And we can also see that the system has optimally decided on a limit over here where the grid purchases are saturated, and that is the optimal value that I told you before to try and save costs on the grid demand charges. So in other words, the system is working just as expected, and we can see that it is all working interchangeably and working successfully. So in conclusion, we have created a hybrid renewable energy model to serve the energy needs of Hermanus municipality with a total of 10.5 megawatts of renewables and 39 megawatt hours of battery energy storage systems, with a renewable energy penetration exceeding our requirements of 50% at 53.9%. We have incorporated 7 EV charging stations successfully, as well as a battery energy storage unit to deliver a reliable, smooth power supply with no unmet electric load. So the levelized cost of electricity for this system is R1.37 per kilowatt hour. And the really amazing thing about this study is that this is a flexible model that can be applied to other towns, so it's really useful for for municipalities, policymakers, and town planners. So in terms of future work, I think I would like to use recorded data for more representative modeling instead of just making assumptions, incorporate the converter costs to showcase real-life costs that are associated with renewable energy systems, and use more relevant parameters, for example, more relevant financial parameters and costs of PV panels and wind turbines. Thank you very much, and you're welcome to contact me for any more questions.
Well done, Stefan, on such an excellent presentation. Can we take questions, colleagues?
Thanks, Stefan. So I imagine on your last results slide that the batteries are lithium-ion.
Yes.
How do you think solid-state battery technology would compare to that?
I'm not too sure. I haven't really evaluated that too much, and the main reason for that is simply because of the cost of lithium-ion as well as the benefits. I think lithium-ion batteries are almost the jack of all trades when it comes to batteries. I think the major concern with them was safety in the past, and we had a lot of uncontrollable fires. So I'm not sure how solid state would compare in terms of a technical standpoint, but I can imagine that it must be more expensive. And in a situation like this where we're looking at an economic driver, I think lithium-ion batteries make a lot of sense. But it's a great question and something to be investigated.
Sure. Thanks.
Thank you. Just more like a follow-up question, Stefan. So sodium-ion batteries are more cost-effective than lithium-ions, but they're not safer than lithium-ions. They explode a lot, the sodium-ions. So we should find along the energy storage literature that more people are investigating sodium-ion batteries than lithium.
Yeah, absolutely. You must also remember that cost is not necessarily tangible. So yes, you can go and buy an off-the-shelf battery here, you can go buy an off-the-shelf battery there, but there's also cost and risk. And in many of these projects, that risk is too high for a sodium battery. So that's why I think it's almost become a standard procedure to incorporate lithium-ion batteries, especially because of the EV market, where a lot of research has sort of pushed lithium-ion batteries. But I think there's definitely lots of new battery sources coming through. It's not like lithium-ion is the holy grail of batteries. You can do some research on vanadium batteries, which are also really impressive, that don't degrade over time, or very, very little degradation. They're just quite expensive, and they're currently sort of in the proof of concept stage. But I definitely think that it's battery energy storage is always something that we want to improve on, and the more energy storage we can harness, the better we can produce our grid networks and energy storage system delivery.
Sorry, on that thought, did you incorporate some of the smart grid principles in your research? Because I heard you mentioning exporting and importing energy in the grid.
Yes, absolutely. So if I actually can go back to one slide, hopefully you'll be able to see this. But you can actually see over there of the consumption. So the AC primary load is essentially just whatever the demand is. So if you're switching on your kettle or whatever, that would be your AC primary load. Your EV charges is pretty self-explanatory, and then your grid sales is 11.5%. So of the consumption, 11.5% of that is actually being sent to the grid. So this is just a showcase for municipalities to understand that it's not all about just, if you have a solar panel, whatever that is producing, you use in your municipality or your system. There's actually the opportunity to sell that back and make a profit. So it goes back to a lot of energy trading, and if you want to learn more about that, I'd highly recommend looking into VPP's virtual power plants, which is the next big thing in terms of smart grid technology. Yeah, cool. Any more questions? Yeah. Okay, cool. Thank you.
Our next presenter will be Given Ndlovu.
I think I might be a bit short here. Hopefully you can hear me. Afternoon, everyone. My name is Given Dlovu from Energy Center, CSIR. I have a background in electrical engineering, and yeah, I'm a candidate researcher with primary focus on power systems. Thank you. Thank you. So, the work that I'll be presenting today is a pilot study that was undertaken in collaboration with ESCOM with the aim of determining or investigating the impact of embedded generators on their distribution system. So just to give a bit of a background, power systems around the world are going through a gradual shift from these centralized vertical systems where power was generated from the top and transmitted down to passive customers, as you can see on our diagram on the left side of it. And now, with the active participation of consumers where they can generate their own power as well as feeding back into the system, we have a decentralized system, as you can see on our right-hand side of the diagram. So this integration of embedded generators into the network system typically reduces power flow on the downstream by supplying the local loads first and also ultimately reducing the system losses. However, while all this is good and well, there are issues associated with this, and some of the issues, as you can see, is the reversal of power flow, excessive voltage deviations. As we know, in terms of solar, that is not reliable that much. In the case of a cloud cover, you can easily lose power from maximum to minimum, thereby altering the voltage. And also an increase in system losses, especially if this generator is not optimally located or coordinated. And lastly, the complexity in cost recovery and fair tariff design, which I'll get to in more details later. Now, as I've mentioned, this project was done in collaboration with ESCOM with the aim of determining the impact of these small-scale generators into their network. And the reason for this particular study was that initially ESCOM assumed that all these generators are actually helping the system by reducing the power flow and ultimately reducing the system losses. However, they are seeing new trends where there are excessive losses which are caused by these embedded generators, but obviously not all of them. Looking into the losses, system losses in distribution network comprises technical losses as well as non-technical losses. So the first one, they are inherent due to the resistance on the impedance of the electrical network. In other words, they are there by nature or by the virtue of the network. And the second one is the non-technical losses, which I think most of us are familiar with. This one can occur due to metering issues or illegal connection or theft of power. But in this case, we're only going to focus on the technical losses. Now, to summarize this slide, these loss factors are accounted for in the tariff design. In other words, when you purchase energy, the losses that are occurring in the network are already included in your charge. By definition, these loss factors, also referred as loss sensitivities, represent the marginal impact of active power injection at a particular node on overall system losses. In other words, to give an example, if I have a generator here and generating a certain megawatt, how much losses can this particular generator cause on the network, or how much losses in reduction can it cause, or how much help can this particular generation, assist? Going forward, these loss factors that we've calculated can be negative and positive. A positive loss factor will mean that that particular generation is actually causing losses on the network, whereas a negative loss factor will mean that it is helping the network, as I've explained initially. Some generators, or initially all of them were thought to be helping, however now the trend seems to be the other way around. I will explain more, I'll get to that. Now, looking at the methodology, there are essentially two methods used to determine these loss factors. The first one being the classical load flow, and the second one being the analytical based. And not to bother with the details, in our study, we went with the first one simply because the power factory or the software that we were using was compatible with this particular one, and it was suitable for our studies. The only downside was that it can be slower, especially when the network is very huge. And the next three slides basically explain how we went about calculating the losses in our studies and the approaches and the philosophies basically. So what is happening here is that we have high voltage network or case files, and also the medium voltage case files. So if you look at the ESCOM network, the transmission normally goes all the way to 32 or even 22 sometimes. But in this case, we're specifically looking at not necessarily transmission, but though the high voltage network was going into the transmission, and it was starting from 22 kV all the way to 400 in most cases. And anything below 22, it was represented as a lumped load. And to give an example for this one, if you look at, for example, the city of Tshwane network and try to locate CSIR, you probably won't see it. And if you do, you're probably going to see it as a single load, which is not accurate, it's just a representation of it. And then the medium voltage case files on network, they mostly start from 22 all the way down to 400. So again, using the CSR example, we have our own network here, which starts from 132 all the way down to 400. So to determine the impact of these generators on the overall network, we had to get a more representative network by merging the two case files, or two case files. So in essence, we had to replace the LUT aggregated load with the detailed model. So in this case of CSIR example, you will take the city of Tshwane, remove that aggregated load, and merge it with the detailed CSIR model to get a more representative network, because that's how it is in reality. So this diagram basically highlights the methodology that we followed. Firstly, we had to merge these case files, ensure that it can converge, and then calculate the loss factors. But in this case, we had to make sure that we get the performance of the network before we even integrate the renewables. So we had to switch the renewables or put them out of service and calculate the loss factors. And thereafter, integrate or put them in service and then calculate the new loss factors. And then after that, analyze. So what's happening here, we know that as I've explained, the integration of these renewables, they actually change the set point of the network. Hence, we had to get the base without the renewables, and then compare to see whether these generators are causing losses or reducing losses. Now, this is more or less similar to what I've just explained. To determine the impact of these generators, we had to create some scenarios, the first one being the base case and the other one being the generator scenario or case. And then the diagram here highlights the actual methodology on the power factory Dix Island. So firstly, you had to select your methodology, which is the classical load flow, and then select injection nodes. So in this case, the nodes where we were injecting power to see the impact, it was the generator nodes, meaning that it's where the generators are connected. And then we had to select the equipments to monitor. In this particular case, by default, the software will select all the elements, including lines, transformers, switches, and everything. But we had to filter and only look at the branches or lines, and also select our desired threshold sensitivity. By this, in this particular case, we went with 10%. So what that actually means is that if you have a generator here and you are injecting power, we want to see the impact of it on the rest of the network, but it wouldn't make much sense to have a generator and then here in Gauteng, like a smaller generator, and then look at Mpumalanga. There might be some changes, but it's very, very minimal. So we had to cap it and say 10% is good enough. Anything below that, we neglect it, because in most cases, if you have a smaller generator here, there might be no change in Cape Town or somewhere far, but we're focusing on the nearby network. So from there, this one, we're basically choosing to evaluate the impact of these generators one by one or all of them at the same time. So in our studies, we did both, where we were evaluating each generator in 10 and also all of them at the same time. But for our studies here, we will only show for demonstration, we'll only show the first one. And then looking at the results and how to read or interpret them. Remember, initially I mentioned that a positive loss factor will mean that that particular generator is actually causing losses or increasing, contributing in system losses, and the negative one will be the other way around. So in this case, as I've mentioned, we had to do the study without the generators and obtained these particular values, and then do the very same study but with the generators in and obtain these. So if you look at generator number 1, or rather node number 1, because this could have been a node, doesn't matter much. If we look at generator number 1, initially we obtained a value of 0.564, and with the generator, the loss factor increased to 0.587, which indicates that the presence of this generator will actually cause or increase losses in this particular branch. And again, for more information, we chose a branch for this. So this is just one branch we are monitoring here, as I've mentioned here. And we are looking at the impact of all these generators on that particular branch. So you can see here that generator 1 as well as 4 and 5 are exhibiting the same behavior. They're actually increasing losses on the system. However, generator 2 and 6 are actually helping as far as system losses are concerned. As you can see here, we had 0.693, and now we have a lower loss factor. And then if you look at another example where at first glance you can tell that we have a lot of negative, and I've said that a negative loss factor will mean that the generators are actually helping the system. So on the base case, these are the loss factors we've obtained. On the generator case, we've obtained these loss factors. Again, if you look at generator number 1, you can see that we have a negative 0.759. However, now we have negative 0.157. So all of these generators are exhibiting the same trend, but what we are saying here is that the loss factors increased in a sense that they are going towards 0 or a positive. If we keep increasing generators here, you will end up with a positive loss factors. So in conclusion here, it means that yes, these generators are helping as far as system is concerned, system losses are concerned, but if you keep increasing generators in that particular area or this region, you will end up with a case where all of them or most of them are actually contributing to system losses. And then in conclusion, as I've said, the integration of this renewable energy, they typically reduce the power flow downstream and helping with the system losses. But these studies, the findings of the studies showing that this is not uniform all over the distribution system. And it is in line with what the ESCOM guys are seeing, these losses. So yeah, while some areas can benefit from these reduced losses, others are actually experiencing increase in these losses. And in some cases where the local generation exceeds the local demand, you will have the reverse power flow, which makes sense. If you have a solar in your house and you keep increasing, you will end up not wanting power from your main utility. You will actually be feeding back eventually. And that can, in some cases, cause losses. in some cases it can reduce, but in some cases where the network is probably constrained already, it can cause losses. So this suggests that although embedded generation can help moderate losses, there's a tipping point beyond which its contribution becomes less effective or even counterproductive, depending on the network conditions and the balance between generation and demand. Therefore, further studies are needed to support the development of a more cost-reflective tariff structure for ESCOM. So, in other words, what the study aims to do is to basically say which generators are causing losses on the system and how do we charge them accordingly, because at the moment they are basically charged the same as if all of them are reducing losses. So for a lack of better word, sort of penalize them for causing more losses on the network by creating a more cost-reflective structure. And then for future work, this study was conducted for just one province in South Africa. So going forward, we'll have to do the study for the entire South African provinces, particularly given that there are regional differences in embedded generation as well as their penetration levels. So yeah, these findings will eventually inform policies and regulatory frameworks for managing technical losses and implementing cost-reflective tariffs. That would be my story for today.
Thank you, Given. Well done on such an excellent presentation. I will take questions. Just two, or maybe one. In that case, I guess your presentation was clear. Well done. Thank you so much. He's saying, oh, confusing. Anyway, our last presenter for this session is Samuel Makobe.
Good afternoon, everyone. I know that I'm the only thing that's standing between you going home and being here. Thank you so much for hanging there so tight. Thank you so much also, Chair, for the platform. My name is Samuel Makobe. I'm a PhD candidate supervised by Professor A.A. Ambushi and Dr. M.P. Swapela from the Department of Chemical Sciences at University of Johannesburg. So today I'm going to give a presentation on spatial and temporal variation in presence, level, and risk assessment of selected polycyclic aromatic hydrocarbons in water and sediment from Klip River, Johannesburg, South Africa. Throughout this presentation, I'll be following the projected overview from the introduction to the acknowledgement. So as we all know, South Africa is known to be one of the most As we all know, South Africa is one of the most water-scarce country or the driest country, ranking the 30th currently in the world, with limited water resources that are unevenly distributed throughout the country. So many communities, especially in rural areas, as well as peri-urban areas, people still struggle to access safe and potable water. So I was very happy this evening, colleagues, this afternoon, colleagues, when I was receiving free water. So some of our colleagues are still struggling out there, especially in rural areas. So we can see that the situation led to what we see, or what is depicted in figure 1, where residents from various regions of this country have to stand in a very long queue to access clean potable water. And then we can imagine others who have to buy water, and then also the situation for our elders who are pension dependent, who has to use that money that was meant for survival to access potable water. Besides increasing number of population that we have in this country, human activities such as the mining operation, agriculture, we're looking at increase in urbanization as well as industrialization, often lead to the pollution of the remaining water systems in this country. And this does not only affect the ecosystem, but it also affects the public health. So to address this matter, our government in South Africa have enacted a National Water Act, Act number 36 of 1998, which ensures that the remaining water systems in our country are treated or are managed with equity and sustainability. This law treats water as a national asset by making sure that there's systems in place to protect the environment as well as human needs. And this is made sure also by making sure that there's enough clean water set aside for basic use as well as ecological protection. Klip River, which winds through Soweto and Lenasia, located in the Gauteng province, as depicted in figure 2, which feeds in the main Vaal. This river, it serves as a vital water source for local communities. We're looking at the livestock around the area, as well as the whole ecosystem. But the presence of anthropogenic activities in the vicinity of this river, such as industrial discharges, we're looking at leaking sewage systems around Soweto, we're looking at irresponsible littering. This might lead to the pollution of this water system. Hence, it is imperative for us to conduct an environmental study to determine the identity and the level of concentration level of emerging environmental pollutants in this river. And then the polycyclic aromatic hydrocarbon, abbreviated as the PHs, these are organic compound composed of 2 or more fused aromatic rings. The origin of these compounds include anthropogenic activities such as industrial discharges, burning of fossil fuel, we're looking at irresponsible littering, sewage discharges, as well as oil spills. These anthropogenic activities, they are pollutants of concern because when they're in contact with animals and human beings, they result in carcinogenic effect, and then some of them, they result in reproductive failure as well as developmental malformation. So 16 of these priority pollutants, as depicted in figures 3, have been categorized as the priority pollutants by the United States Environmental Protection Agency. When in contact with human beings, they result in carcinogenicity, neurotoxicity, as well as genotoxicity. And then when these compounds are in contact with aquatic animals, we see developmental malformation, acute toxicity, and over time, this leads to a species extinction. And then when they are in contact with the crops, we'll see the reduced crop productivity, salinization of the soil, as well as biodiversity loss. Then the aim of this research was to determine the identity level, sources, as well as the toxicity of these contaminants in water and sediment. The specific objectives include evaluation of concentration level of these pollutants into different seasons. We also wanted to optimize and extract these pollutants in water and sediment. In water, using liquid extraction method, and then in sediment, we used microwave-assisted extraction. We also want to know if our method were validate, so we confirm the accuracy of the method applied in extraction of these compounds in water using the spike method, and then in sediment sample, we use certified reference material 104 for sediment sample. We also want to further analyze our sample using gas chromatography flame ionization detector, and then we apply a new study, environmental toxicity study, because we don't want to tell people that the water is contaminated, then we leave it like that. People want to know how does that affect them. So what we did, we introduced the new study to evaluate the ecotoxicological effect of the whole water and sediment using the zebrafish embryo development test. Figure 4 shows the exact location of the sampling site where our samples were collected in Soweto and Lenasia, in Johannesburg. Of course, we have to do the hard work, go there, collect the water and sediment. I bet this is the very bad job. No one will like it, but over time we get to be in love with it, colleagues. Then for sample preparation, prior to collection of our samples, water and sediment sample, we evaluated the measurement, we evaluated the water quality parameters to understand the quality of water. Then after that, we collected water in those brown amber glass bottles, 2.5 liter, and then the sediment samples were collected in transparent glass jar. Then we took our samples in a cooler bag to the laboratory. When we get to the laboratory, we have to refrigerate our water sample at 4 degrees Celsius, and then we freeze-dried our sediment sample using a bio-based freeze dryer. Then we grounded them into a fine powder using an agate pestle and mortar. Then we sieved them and analyzed them in a microwave-assisted extraction using 30 milliliter acetone and hexane in 1:1 ratio. Water samples were then subjected Water samples were then subjected to Buchner funnel filtration. Then we performed a simple liquid-liquid extraction by mechanical shaking using 3 times 60 milliliter of dichloroethane. Then for method validation, we employed certified reference material 104 in sediment sample, and then we used the spiking method to confirm the accuracy of the extraction method in water. Then extract of water and sediment sample were then collected and analyzed using an Agilent Model 7820A gas chromatography flame ionization detector under the following depicted optimized operating condition in table 1. Then we performed the toxicity test. This work is very difficult. At some point, you are an environmental analytical chemist. At some point, now you become a fisherman. So yes, we have a facility in Northwest. We call it a bioaquatic national facility in Northwest University, where we have our zebrafish embryo and then the zebrafish who are confined in different housing tanks. So we choose this fish, zebrafish, because of it shares over 70% morphology with human beings. This implies that upon exposure to contaminants, the way they respond has a probability over 70% similarity with human beings. So this zebrafish embryo were collected, and then we separated them into male and female into 2-to-1 ratio. So the same as human population, more women than men, right? And then after that, colleagues, we are able to separate the two. So the one that has a belly, which means it's carrying eggs, right? That's a female, and then you have a male at the top. Then we took them, we inserted them into the ice pond tank for mating purpose. Male at the top, female at the bottom. Then we switch off your light. You come tomorrow, upon separation of this net, we separate them in this net, then we take out that net, and then they start mating. After mating, we collected the zebrafish embryo at the bottom of an ice pond using the petri dish. Then using this microscope, we were able to separate fertilized embryo from an unfertilized embryo. Then we took fertilized embryo of the same cell stage, then we evaluate endpoint such as mortality, hatching rate, and malformation. We were able to detect the presence of 16 pHs, including 4 internal standards, using gas chromatography flame ionization detector based on different retention times, as depicted by the chromatogram in figure 8. Then we used the internal standard calibration method to construct calibration curves for the concentration determination of each pH. So in figure 9, we're showing an example of calibration curve obtained for fluorine. And then the following analytical figures of merit were determined. Coefficient of determination, which shows good linearity. We also evaluated limit of detection as well as limit of quantification. We looked at percentage recovery of our method, which shows good and acceptable recovery. We were also concerned about the precision of our method, and we evaluated standard deviation as well as percentage relative standard deviation. Figure 10 shows an example of analysis, example of chromatogram obtained from an analysis of extract of sediment sample. In the first picture showing the presence of pHs, and then the other figure is the chromatogram obtained from analysis of an extract of water sample showing the presence of pHs. The presence of peaks other than that of pHs here, it indicates the sample complexity of our sediment compared to water samples. Then we were able to determine the concentration of these pHs, polycyclic aromatic hydrocarbon, at each site, also the combined concentration of all of them during the low flow season and during the high flow season. So we were able to also note that the concentration level of these contaminants were higher during the low flow season compared to the high flow season. And then we were also able to detect sediment site with elevated level of pHs, such as site 7, a reference site in the low flow season. And then we've seen the observed, and the lowest was recorded in site 4. And then when we go to the high flow season, we have observed temporal variation in concentration level of this, which might be also attributed to different activities that were happening at that particular site. So we observed that the concentration changes. Elevated concentration was in site 6, and then the lowest was observed in site 3. And similarly, we also evaluated their concentration level in water during the low flow season and high flow season. So we also able to detect site with elevated level of pHs in the low flow season and the high flow season. And then we also noted that the total concentration collected in all sites during the low flow season were higher compared to the high flow season. But it's imperative to note that the highest concentration of these pollutants were found in sediment sample compared to water samples. Then we further conducted the toxicity study by exposing our embryos to sieve sediment sample that we collected in the river. Also, we exposed them to water sample, and the test was conducted under two controls: the negative control indicating the medium, clean medium, and the positive control indicating medium with toxins. So we first monitored the hatching rate, because our zebrafish embryo were expected to hatch naturally at 48 hours post-fertilization, indicated by the red dotted line. If you look at the embryos exposed to the negative control, we've seen normal hatching rate above 50%, but when you expose them to the medium with toxin, hatching rate was affected as it was below 50%. If you remember the site with elevated level of pHs in sediment sample, site 7 during the low flow season and site 8 during the high flow season. So you can see that embryos at that particular site, at those particular sites, did not hatch at 48 hours post-fertilization. Did not hatch at 48 hours post-fertilization, and hatching started at 72 hours post-fertilization. This means that the presence of pHs at elevated level negatively affect or delay the hatch rate of zebrafish embryo. And the same trend was observed in water sample, but in water sample were not negatively affected compared to the sediment sample, showing the presence of less pollution, pH pollution in water samples. We also evaluated the mortality of zebrafish embryo under two control. So we can see that in a clean medium, we have less mortality of less than 10%. And then when we expose them to the medium with toxin positive control, we have around 30% of our embryos were dying. If you look at site with elevated level of pH, site 7, during the low flow, and site 8, we can see that the mortality was increasing with exposure time, meaning the more you expose the embryos to those sites with elevated level of pHs, they were dying with time. And then at the end of our test period, it's also imperative to note that all of our embryos exposed to site 6 and site 7 had died. So the same trend was also observed in embryos exposed to the water sample, but the level of pollution in water sample was so less, so you can see the mortality was less, which means some of our embryos were surviving in water samples. And then we went further and evaluated the total malformation overall. So malformation over 10% was regarded as significant, which means it's site of concern with regards to developmental characteristics of this embryo. So we can see in the sediment sample, only site 9 during the low flow season was insignificant. So all other embryos exposed to these sites of sediment sample were experiencing different developmental malformation. And if you look at our sites of interest, site 7 and site 8, you can see over 80% malformations were observed in those embryos. Similar trend was observed in water sample, but because water samples were less polluted compared to sediment sample, some of our embryos didn't exhibit significant malformation from 24 hours to 48 hours post-fertilization. At 72 hours post-fertilization to the 96, that's when they started exhibiting significant malformation. Then we went and evaluated the heartbeat per minute of this zebrafish. We want to know if ever the heartbeat was not affected. So we exposed them to different media, including negative control and positive control. So we can see in a clean medium, our embryos are surviving, but in medium with toxins, our embryos are getting affected, especially in sediment samples. So I've also included a video, guys, that we made using a dinoscope showing you that how we did integrate that part and determine the heartbeat of the zebrafish embryo. We went again and determined the blood flow activity, because if the blood is not moving, they'll get to die. So we can see in sediment sample, the blood flow was affected, and I've also included a video there showing how we selected a portion in zebrafish and determined the blood flow circulation of those zebrafish. And lastly, we evaluated the burst activity. We want to know the movement, because if this site is polluted, you need to move to the other side. Also, this is the survival skill. So we can see that in the negative control, our embryos were able to move effectively. But look at this, in certain sites, our burst activity, the movement of our zebrafish embryo were really affected. So I've also included a video here. Every time it gets to a certain peak, we're able to calculate the burst activity, the movement of this zebrafish embryo. So I hope colleagues, you can see them moving there. We use the stethoscope, remote control, and we're able to detect those things. All right, in conclusion, we're able to observe that anthropogenic activities present in the close proximity of the river leads to high pollution in water and sediment sample, leading to the presence of pHs at elevated level. We're also able to determine that the low flow sediment and water sample had high concentration level of pHs compared to high flow. And then we're also able to see site with elevated level of pHs during the low flow and high flow season in water sample and sediment sample. And then from the toxicity test, we're able to also see that highly polluted site with pHs leads to low Hessian rate, high malformation, and then high mortality. So in future, we want to look at other emerging contaminants, such as the inorganic, especially the inorganic part, such as potentially toxic element. We can look at pharmaceutical microplastics, as well as any other endocrine disrupting compound. So I'd like to acknowledge my supervisors, Prof. Wepener, also from the Department of Zoology in Northwest. There's the guy responsible for the zebrafish facility. And then the analytical chemistry group at UJ, the funders, CSIR, NRF, Water Research Commission. And lastly, it's imperative to know that this work has made a significant impact, scientific and public contribution. So we are able to publish part of this work sediment analysis in February. And then this work also has received a wide range of media coverage. So if you want to follow the discussions that were done in different platforms, you can click on those links. I thank you all. Ke a leboga.
Well done, Samuel. Any questions for Samuel?
Thank you. So I want you to know, the test was done with zebrafish.
Sorry?
The test was done with zebrafish on the variety of issues on the Klip River. So what other fish are native to the Klip River, and how are they managing in that environment at the moment?
We're not particularly looking at different kinds of zebrafish, but we know that the genome of zebrafish is highly conserved in animals, also in human beings. So particularly when I was saying that prior to extraction, prior to sampling, we look at water quality parameters. So such as total dissolved oxygen, total dissolved oil solid, you also look at the pH of the water and all that. So the total dissolved oxygen was very less, showing that the aquatic animals were really affected, which means it's hard for them to breathe. And we also noted a high concentration level of the solids. In addition to that, a lot of people who are fishing in selected sites that we considered were also complaining about the amount of fish that they were getting, because those people have been staying there for a very long time. So they used to go there at this particular time and get a lot of fish. But nowadays they're not getting anything. So which means that the survival rate of those aquatic animals at those selected sites are highly affected. I hope I answered your question.
Yeah, thank you.
Thank you, sir.
Thank you, sir. Any challenges that you face while conducting your research?
I faced the challenge. Okay, the first challenge was with instrument, because I was concerned about the sensitivity. So, and then we had our instrument break down GCMS, because I wanted GCMS or TOF-MS, looking at the sensitivity. Then because of the research has to move, then I used the GCFID. Secondly, because of I'm wearing glasses, then during the during the mating and all that, remember having to walk in the lab, and then it's dark and then it's steamy. So you get all of that. It's like driving a car when it's raining. So my glasses were very, 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 very harsh. So I couldn't see anything. But at some point, obviously, you have to stay strong and then conduct the toxicity test. Thank you so much.
Thank you, colleagues. Can we just give one last round of applause to all our presenters? Thank you, colleagues. We are now heading towards the end of the session. I just have a few announcements to make. For tomorrow morning, I've been asked to ask all the colleagues to make sure that they are all here tomorrow by 8:30 so that we are all seated for the CEO's keynote address tomorrow. And then with that said, I would like to again thank all the presenters for the excellent work that they did today. Thank you to you all, ladies and gentlemen. You have been such an amazing audience and you made this session very engaging and very interesting. And for that, I thank you. Last one, as we leave here, can we all head towards the posters and the exhibitions? And then with that said, I call this session to its close. Thank you.